Good morning, champions. Let me see if at all I am visible. Yes, I am. Good morning, Jia. Good morning, Shalom, and good morning, Ganesh. Also, Shivanand. Hello. Very good morning. Good to see you all over here. So, my dear children, quickly send this link to all your friends out there who also wants. Good morning, Sanjeevani. So, I'm really excited to have you guys over here. I hope I didn't disturb your morning. So, children, make sure that you, send this link to all of your friends out there. Good morning, Lakshmi, Shalom, and Jia, everyone out there. I hope you're doing great. So, are we ready to do the 1-marker questions? So, today, when I say that I am going to start, I believe that it might end up at 12:30. 4 hours is the maximum that I'm taking for MCQs. Now, I understand that you do not have much time, which is exactly why I'm telling that. But yes. Ma'am, okay. Ma'am, have you had tea or coffee? So, I do not have either of them usually. So, I had water. Thanks for asking though. I hope you guys had your breakfast and everything. So, my dear children, this particular video would be having questions from your MQP, model question paper. So, we will be seeing few repetition of questions, model question paper, because we have solved it already. And then we would be solving some questions from the question bank as well, and all of it. Okay? Yes. So, let's go. I hope we are all ready. And ma'am, you're looking cute. Thank you so much. That's the best compliment I got in the morning. And you guys are also really cute. Yes. Let's go ahead then with the first question that we have, and that is from the chapter Sexual Reproduction Flowering Plants. We'll go chapter by chapter. And Ankit, good morning. Okay. So, the first question that we have over here, I believe everyone should be able to solve this who have solved MQP with me, and that is statement number 1. Pollination does not guarantee the transfer of right type of pollen. Statement number 2. Compatible pollen-pistil interaction guarantees double fertilization. What do you think is the answer for this? You can give me the answers like this. 1A, 1B, 1C, 1D. Whatever is this, you have to tell me. Ganesh Patil says C. Okay. What about others? So, those of you who have completed MQP with me, I expect answers from you guys. Shalom says A. Jia says C. Ma'am, you're not feeling well. I am doing absolutely great in the morning. Okay, Ankit Thakur also says C. Ganesh also says C. So, I see maximum votes are for C. And what is C? C says statement 1 is correct, but statement 2 is incorrect. And you guys are absolutely correct. Awesome. Perfect. Okay. Hello, Embryo and Bhairu and Sanjeevani. Lakshmi, okay. So, I see confusion between A and C, which is exactly why I'll just explain to you quickly. Pollination does not guarantee the transfer of right type of pollen. Yes, it does not. For example, I am a plant over here, there is another plant over here. It can be of another species, but still pollination can happen. It can happen, but obviously we are of both two different species out there. So, it does not it does not guarantee any transfer of right type of pollen. That is correct. Now, this is where most of us get confused with compatible pollen-pistil interaction guarantees double fertilization. So, you have your stigma over here, style over here, and ovary over here with your ovule, my dear children. And inside that, you would be having what? The female gamete. The female gamete is right over here. Suppose when I say pollen grain lands up over here, and all the interactions are totally fine, it forms the pollen tube, right? And you believe that this pollen tube might go in there and do the double fertilization process because it is compatible. No, listen to me. Multiple pollen grains can land on the stigma at the same time. They can form multiple pollen tubes as well. Some of them they can go ahead, some of them might not go ahead. So, I cannot say that compatible pollen-pistil interaction will guarantee double fertilization. That is not a guarantee at all, which is why your answer for this question is going to be C. Statement 1 is correct, but statement 2 is incorrect. So, those of you got your answers correct. Those of you did not get your answers correct. This is how we learn. Yes. Going ahead with the next question that we have, statement number 2, again from MQP2. Hello, Bhairu. I hope my dear children are having a very good morning. Yes, and the next question goes like this. Statement number 1. Apomixis is the production of seeds from unfertilized ovules. Is that so? Now, statement number 2. Embryos produced from apomictic seeds are not generally identical to the parent plant. What do you think so? Again, everyone, MQP guys, come on. 2A, men is maths life. Pradyun, it would be towards afternoon is what I believe. Yeah? Yes. 2A, 2A. Ankit Thakur also says 2A. Shalam also says A. What is A over here? Statement 1 is true, but statement 2 is false. Is that so? G also says 2A. So since most of my children are saying A, I have to go with A only. A is true and 2 is false. Okay. Apomixis is the production of seeds from unfertilized ovules. How can you get seeds from unfertilized ovules? Only fertilized ovules become seed, right? But yes, apomixis is an exception where unfertilized, without fertilization itself, the ovule will be transforming into a seed. Why? Because over here, the egg which is there, it is not haploid. Usually egg is haploid, it waits for the male gamete to come, such that haploid comes together to form diploid thing, right? But over here, the egg is already diploid in nature, which is why it does not need any of the male gametes out there. And that is how basically it can form a zygote by itself. There are multiple other ways also to do apomixis. This is one such way where you are getting a seed without fertilization. So this is correct. Now embryos produced from apomixis seeds are not generally identical to the parent plant. This would be wrong, right? Right? Why wrong? Listen to me. Here it is written, they are not generally identical. Suppose I am the plant which is being formed from here. Listen, I just look like directly like my amma only. I do not have any choice. Only my amma was involved over here, right? No papa is there, only amma. So obviously whatever genetic material is out there, I'll be getting all of that. Which is why I am going to be genetically identical to my parent plant. It's written not identical. So that is obviously not true, which is why the answer for this question is going to be A. And you guys were correct. I did not see any wrong answers out there. Awesome. Okay, you have understood the assignment. Now question number 3. Even in the absence of pollinating agents, seed setting is assured in. This would be the last MQP question. After that, there is question bank questions. Even in the absence of pollinating agents, seed setting is assured in. Disha is also there. Hi Disha. Sanjeevani says C is the answer for this question. Disha also says C. Shalam also says C. Oh my god. Ankit, Ganesh, Jia. Oh my god. Everyone is saying Rahul. Hello. So everyone is saying C, which is why I'll go with C. Commelina. Why Commelina, Baba? Because Commelina is a Commelina is a plant that has both type of flowers, Clistogamous and Chasmogamous. Clisto means what? Clisto. Over here CL for closed. Always remember. So this is basically closed type of flowers where you have the female part, you have the male part. So anyway, without any pollinating agents, it will go up there, do the pollination out there, everything will be done, everything is set out there. Okay, what are the other examples for this? Remember, Commelina, Oxalis, Viola. So I remember it as cow. So cow with a V over here. Those are the examples for this. Make sure you know about it. Apart from it, they also have open flowers. If they have open flowers, that means what? Autogamy can happen. Okay, since they have both the male and the female part, gynoegamy can also happen, xenogamy can also happen. Please know about these terms as well. So when they give you a question like this in your MQP, you should be knowing any related thing about that. Okay, so go through every single thing. So now we have done with the, the MQP question, and now, yeah, this is the clistogamous flowers. They are mostly found towards the root. We will be going ahead with the question bank questions out there. Select the correct sequence of anther wall layers. Come on, my champions. Select the correct sequence of anther wall layers. So over here you have all the things. You have to choose what is the correct answer. Please give me an answer by like 4, A, B, C, D. Put the term 4, okay? Otherwise I will not understand which question you're solving. Okay, Shalam was the first one. She says it is 4B. Okay, Sanjeevani says 4B again. Ganesh says 4B. Okay. So I saw a different answer. Ankit says it is D. Okay. Disha also B, and yeah, most of them, Rahul also say, Jia, Rahul, everyone is saying B. B is what? B is what? Epidermis, endothecium, middle layer and tapetum. There's an easy way to remember it, okay? Listen to me. You might have heard about ENT doctor, right? You go to the ear, nose, throat doctor if at all you have any of the problems up over here. Similarly, this is EMT doctor, okay? E for epidermis, then you have endo, then you have middle layer, and then you have tapetum. Now a question that is out of the box. So your answer for this question is definitely going to be B. Now you tell me, okay guys, you tell me one very important thing. Out of these 4 layers out there, which ones are there for involved in dehiscence of anther? Which one is involved in dehiscence of anther? Come on. Dehiscence of anther. Which is going to be involved in the dehiscence of anther? Dehiscence of anther means breakdown of anther to give out the pollen grains. endothecium. Akhil is also there. Akhil says the answer for this question is D. Dear, I hope you have got the answer now. Children, I am asking about dehiscence of anther. Wrong, 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 wrong. Think and say, think and say. Do not just look at others and give the answers. Epidermis, okay. Disha says endothecium and Sanjeevani says epidermis. Shalam says endothecium, okay. Listen to me very carefully, okay? There are 3 layers which are involved in the dehiscence of anther. Dehiscence means breakdown of anther. So those are EEM, these 3 layers, epidermis, endothecium, middle layer. Since they are towards the outside, they are the ones that have 2 roles. One is to provide protection to the anther, second is to help with dehiscence of the anther such that the pollen grains can be released. Okay, this is super important, EEM. Now what about tapetum? Tapetum is a whole different layer. It is just around the sporogenous tissue. So this is a sporogenous tissue where your pollen mother cell or your microspore mother cells are there. They are going to form the future microspores and later pollen grains, which is why they need nourishment. Who provides the nourishment? The cells of the tapetum. The cells of the tapetum are going to provide nourishment, and you should know that they are having too much amount of cytoplasm. They have a dense cytoplasm, and not just that, they have a unique character that is having a binucleate nature. Binucleate nature means they have 2 nucleus in them. So why do you think that they have 2 nucleus? Karyokinesis took place, nuclear division took place, but cytokinesis didn't take place. Cell did not divide. That is why you have what? 2 nucleus out there. Do not mess it up. Tapetum is for nourishment. It has 2 of these features. Do not forget it. Okay? Shalam, tapetum is for nourishment. Exactly. Akhil nourishes the developing. Exactly. So my dear children, I hope the next time you get any questions related to this, you are not going to mess up. Right? Yes? So everyone who thinks that it is perfectly good to go ahead, just give me a thumbs up. We'll go ahead. And I hope you guys have shared the video link with all of your friends. I saw in WhatsApp poll there were around 300 children who voted for 13th morning live. And now I just see few children watching this video, which is actually sad. Aishwarya Patil. Okay, fine. Thank you guys for everyone for giving me the thumbs up. Let's go ahead with the next question. Yes. Okay, select the correct sequence of events in microsporogenesis. What is the correct sequence of events that is there in microsporogenesis? Sporogenous tissue, then microspore mother cell, then microspore tetrad, then microspores. What do you think is the answer for this? Shalam says the answer is A, 5A. Akhil also says A, Disha A. Okay, Jia also says A. Aishwarya says A. Sanjeevani says A, Ankit Thakur, everyone is saying A, which is why you have to lock it up. Sporogenous tissue, microspore mother cell, microspore tetrad and microspores. This is going to be your answer. Let me just tell you, for those of you who might not remember it well, listen, you have the microspore mother cell, okay? This cell is there inside the sporogenous tissue. All the cells which are there, they have an ability to divide. Unlike in the case of ovule, in the megasporangium, only one cell is going to divide. Here, all the microspore mother cells or pollen mother cell has the ability to divide. So, what are they going to undergo? They will undergo meiosis 1, they will get 2 cells. They will undergo meiosis 2, they will get 4 cells. And this 4 cells basically says that, oh my God, I am not going to be away from anyone. I will be together. So, this is microspore tetrad. The microspore tetrad is being formed. Then later, these microspores which are there, my dear children, they are going to go ahead, be individual microspores, and these individual microspores will later give rise to your pollen grain. So your answer will be sporogenous tissue. Inside that you have this. Inside that basically it undergoes division to form microspore tetrads, and then finally you have microspores. So your answer for this question is going to be A. It's an easy question, but yes. Next question. From the statements given below, choose the option that is true for a typical female gametophyte of a flowering plant. What is true is being asked. Ma'am, is tomorrow biology? No, dear. Biology is on Monday, but the reason I am taking it now that some of us do not have mathematics, okay, and we directly have biology, and we have few days left, and within this few days, I don't think on any other day I can take MCQ live, because that would not be of too much importance then, because you would have to complete your syllabus, right? So that is exactly why I'm taking it today. And Sanjeevani says the answer for this question is 6A. Okay, Ankit Thakur also says it is A. Shalam says A. Akhil also says A. I hope, children, you are trying and you're telling. Yes, what is A over here? A means both A and D are true. Rest, nothing is true. Hello, ma'am. Hello, Numan. Children, are you sure it is A? Think about it. Read the question properly. You have to read the question carefully and then only answer. Listen, we might be messing around. I do not want you guys to mess around. Hello, Manjunath. Good morning. Ma'am, it's C. Exactly. Shalom also now says it's C, and Sanjeevani also have rectified that. C is what? A and B. Let me check. So you have been asked what is true. It is A, and true about what? Female gametophyte. What is female gametophyte? Female gametophyte is basically your embryo sac. This is nothing but the embryo sac. Now listen to me carefully. When I say this embryo sac is there, how many cells do I have at the top over here? I have 3 cells. Okay. How many cells do I have over here? I have basically, again, these are 2 synergids and we have 1 egg cell, right? And what about the other cell? This is one big cell out there, and this one big cell which is there, it has 2 polar nuclei. These are nuclei out there. Okay, now listen to me. It is 8 nucleate and 7 cell at maturity. Let me see. 1, 2, 3, 4, 5, 6, and this one is the 7th cell. But nucleus, how many do I have? 1, 2, 3, 4, 5, 6, 7, 8 nucleus is there. So 8 nucleate, 7 cell at maturity. This is absolutely correct. They can twist and turn and give you the same question. They can put 7 nucleate and 8 cell condition. Do not get confused. Make sure you read the question carefully. It is free nuclear during the development. What do you mean by that? Free nuclear during the development. So basically, one megaspore which is there, it is the one that gives rise to the whole embryo sac. Okay, one megaspore. So this one megaspore which is going to be formed, my dear children, what it undergoes is that it has its own nucleus. Suppose this is a cell and it has its own nucleus. This nucleus is going to undergo division. First division into 2, again into 2, again once more. How many times? 3 times division happens. And all of these times, my dear children, if at all you see, the nucleus is getting divided. Okay, it's free nuclear division. And after the nuclear division takes place completely, after 3 rounds of nuclear division, then only cell wall is going to be formed around each of them. This is very, very important. So this type of division where the cell wall is not formed immediately, I'll say it's free nuclear division. And that exactly happens during development, which is why I'll say B is also correct. Now, it is situated inside the integument but outside the nucleus. So if at all you have seen the structure of the ovary, this is the ovary, this is the ovule. Ovule feels very cold, which is why it wears a sweater, that is integument. Again, one more sweater is there, outer integument. Now, if this is ovule, my dear children, it will be having certain cells within it. You say that they are the cells of the nucleus. And within this, you have the embryo sac. So, embryo sac, guys, innermost thing out there. Outside the embryo sac, what do you have? You have the nucleus. Outside the nucleus, what do you have? You have the integuments. So, it is situated inside the integument, but outside the nucleus. That would be a wrong statement to say, which is why exactly inside the integument, right? So this is also wrong. Now, it has an egg apparatus situated at the chalazal end. What do you mean by egg apparatus and what is a chalazal end? Listen, there are 2 ends, okay? In the ovule which is there, there are 2 ends. The area where there are no integuments joining, there is a small pore in between. You call this as micropyle region. What do you call this region? Micropyle region. And the opposite of micropyle will be known as the chalazal region. Always remember, everything important, okay, everything important happens in the micropyle region only. So in this case, this is your egg. Egg is important. That means the micropyle region is over here. What about the antipodal cells? They are not too important, which is why this is going to be the chalazal end. Always remember that. Here also, when I say that one megaspore mother cell is going to divide and form a megaspore and things like that, everything happens towards the micropylar region only. Always remember that. Okay? Cool. So this particular statement is going to be wrong. You just have A and B, which is why your answer is going to be C. I hope this is clear. Ma'am, which part of the embryo is diploid or only haploid? It came in our test. More worried about maths than bio. Is that so? If at all that's the reason, please go ahead and you go do mathematics, because I do not want you guys to be worried about anything. And we'll come back later. You can watch the 1 marker videos out there, right? That would work, I think. Yes. Tanu, 5 marks, important questions, ma'am. Yesterday I gave you, dear. Please go check it out. Okay. Now, which part of the embryo is diploid? Now, listen to me over here. So, every cell over here is haploid in nature. Antipodal cell haploid, this cell haploid, all the antipodal cells are haploid. Here the synergids are going to be haploid, the egg cell is also going to be haploid. Now this particular cell that you see over here, my dear children, this is the largest cell that we have inside the embryo sac. This is known as the central cell, and central cell has two nuclei. One nuclei is haploid, another nuclei is also haploid, which is why n plus n, it becomes 2n. So this is the only cell inside the embryo sac which is going to be diploid in nature. Rest everything is haploid. I hope that's clear. Yes. So my dear children, if at all it's clear, can we go ahead? Just give me a thumbs up and I'll understand that everything is clear for you guys. Perfect. Shall we go ahead? Perfect. Thank you, Shalam. Thank you, Sanjeevani and Numan. Okay. Now, what do we have? Ayo, over here I had the, okay. So this is how the nucleus divides. Okay. The nucleus, free nuclear division, this. 3 times mitotic division happens. Mitotic, do not forget it. And then cell wall forms, and then 3 of the cells antipodals, 2 of the cells synergids, 1 cell out there is known as the egg cell. This is known as the egg apparatus in total. Okay, and over here you have the 2 polar nuclei, and that's all. Now going ahead with the next question out there, identify the correct sequence of arrangement of cells in the mature embryo sac micropylar end. Now, did you have a doubt? Rajesh, please ask me, dear. Okay, Ganesh says the answer for this question is 7A. Rajesh, ma'am, my mistake, I chose reservation category. Okay, Gaganshi, hello, good morning. 7A, Shalam, Numan, Ankit, Ankit and Sanjeevani, Gia, everyone is saying A is what? This. And Rajesh, I do not think I could help you as of now because they have no updates about the other editing window and things like that. Previously you had it and I suppose it's closed. If you're talking about KA, if there is any update, I'll definitely let you know, dear. Okay? Yes, Tanu is also here. Tanu, hello. And Tanu says, Tanu has a different answer. Okay, anyways, let's see. When you're talking about arrangement of cells in the mature embryo sac's micropylar end, that means from here you have to go at the top. In the micropylar end, I told you whatever is important comes towards this particular end, micropylar end. So first thing that you have is synergids. Above the synergids you have the egg cell. Above that you have the central cell. And at the end you will be having the antipodal cells. Always remember that. This is super important. Okay. Amiruddin, hello. Good morning, Ankita. So this is your answer for this question. Your answer is A. I hope you have got it. My mistake I wrote. It's totally fine. Okay, perfect. I do not need to ask you whether you understood because it's an easy question. So let's go ahead. Now, statement number 8. Oh, sorry, question number 8. Statement number 1. In Viola, both autogamy and geitonogamy is prevented. Okay. Statement number 2. Geitonogamy is similar to autogamy since pollen grains come from the same plant. What do you think is the answer for this question? You do not have to be sorry, dear. It's okay. It's totally fine. Chalo. Let's go. In Viola, both autogamy and geitonogamy is prevented. Geitonogamy is similar to autogamy since pollen grains come from the same plant. C. Shalam says A, and Tanu says C, and Gia also says C. C is what? Statement 1 is incorrect, but statement 2 is correct. Okay, most of you guys are saying C, which is why I should be looking and probably hooking on to C. Let me see. In Viola, both autogamy and geitonogamy is prevented. Viola is a plant that has clistogamous as well as chasmogamous flowers. That means the open flowers. Suppose this is an open flower out there. It has the female part. It has the male part. Autogamy definitely can happen. What about geitonogamy? Geitonogamy happens between 2 flowers of the same plant. From here to here also it can happen. So here it is written both, it's prevented. So obviously this is going to be wrong. Now statement number 2, geitonogamy is similar to autogamy. Geitonogamy is similar to autogamy since pollen grains come from the same plant. Yes, if you talk genetically, it is definitely similar because anyway, my dear children, from here to here if it comes or from here to here if it comes, is the same thing only, same plant producing same type of gametes. So genetically, it will be similar. Okay, so which is why I'll say statement 1 is incorrect, but statement 2 is correct. Your answer for this question is definitely C. Yes, only geitonogamy is prevented in Viola. Exactly, they are clistogamous flowers and also, only geitonogamy is prevented. What? No. Gynogamy cannot be prevented. How can it be prevented? Pollination is still happening between two flowers of the same plant, right? So gynogamy is not prevented. Nothing is being prevented over here. Remember, in cow, chasmogamous and clistogamous plants, that is Commelina, Oxalis and Viola, that is what I call it, cow. Listen to me. Anything can happen. Autogamy can happen. Gynogamy can happen, xenogamy can also happen. Now, a question comes, if at all you're just talking about the clistogamous flowers of these plants, then in that case only autogamy can happen. In that case only autogamy can happen. Okay, so yeah. Okay. Bye ma'am, I'm doing math. No issues dear, go ahead. Okay. So your answer for this particular question is going to be C. I hope that works. Now going ahead with this question. Now those of you who knows NCERT really well, who have went through whatever we have learned before, might be able to give the answer for this particular question. The following picture is an example for what? Multicarpally apocarpus gynoecium, multicarpally syncarpus gynoecium, monocarpally gynoecium, apocarpus gynoecium. Gia says the answer for this question is B. Shalom also says B. Sanjini also says it is B. B is what? Tanu also says it is B. Multicarpally condition syncarpus gynoecium. And does anyone know the name of this particular plant by any chance? Nitin, hello. Nitin also says the answer is B. Ankit also says it is B. B is multicarpally. So do they have multiple carpels as in multiple female reproductive system? Obviously yes, they have it. That is what you can see, this. Syncarpus gynoecium means basically they have stigma and style separately, but their ovary is fused. So when we call this condition when the ovary is fused, I'll say that this is syncarpus gynoecium. And that is exactly seen over here. Papa, where is the answer? Exactly. This particular plant that you see over here, Sanjeevani gave me the correct answer. It is papaver. So please know the example for this. This is super important. Similarly, apocarpus condition means they have multiple carpels and the carpels are not fused. They are all having individual ovary. Example for apocarpus. Any idea? Example for apocarpus. If you have the answer, let me know. Mishalia. Exactly, Shalom. The answer would be Mishalia. You know, Mishalia is champak. It smells so good. You Malayalam may say chembagam. Okay, so always remember that also. Anything can be asked anywhere. Kavita, Mishalia, exactly. Shalom, Sanjeevani, and everyone is correct. Okay. Okay. Now Mishalia turned into Michelle also. Okay, this is how it looks, by the way, and yeah. So this is your papaver, and this is what I was talking about. They have their own stigma and style, but the ovary, my dear children, will be fused somewhere. That is why syncarpus condition. And this is the picture that is given in your NCERT for apocarpus condition. They should have given an easy picture. By the way, it looks like this. Okay, so they have their individual stigma and style and everything out there. And you know, this is inside this particular flower. I am pretty sure everyone in their life might have seen this. Okay, this is known as Mishalia, or you call it champak. Okay, champak, it smells so good. Fine. So how many of you have seen this? Give me a thumbs up, and how many of you understood it? Please give me a thumbs up first. And then we'll go ahead with the next question. Yes, ma'am. Exactly. Perfect. So let's go ahead. Do not forget the example. And now we go ahead with the next chapter, that is human reproduction. I saw, ma'am. Exactly. We all have seen, right? Yes. Sometimes your NCERT make things very complicated. Yeah. Now it's... Okay, it's Zaiba. Hello. Okay, now let's go ahead with human reproduction chapter. I hope sexual reproduction, the questions are clear to you. And now obviously I cannot include so many questions out there, which is why some chapters I've taken some questions out there. Yeah, so that it gives you, help you with something at least. And now I have the first question right over here. This question was solved in the MQP. Please everyone try. The secondary oocyte formation from primary oocyte occurs in which of the follicle? The secondary oocyte formation from primary oocyte occurs in which of the follicle? Come on. 1C is the answer. Sanjeevani says 1C. Okay. What about others? Haiba says also it's C. Okay. Tertiary, that means C. Nitin also says C. Okay. So since everyone is saying C, I'll lock it up. Tertiary. Secondary oocyte, my dear children. So listen, you have the primary oocyte. This primary oocyte will divide unequally, giving one polar body, and your secondary oocyte, right? This again divides unequally, gives you second polar body, and finally you get the ovum. It was diploid over here, it becomes haploid here, and it is still haploid over here also. Now you have been asked, this primary oocyte which is there transforms into secondary oocyte in which of the follicle? So the primary oocyte initially was just covered by a group of cells, and you call this whole thing, the cells along with the primary oocyte, you call this as the primary follicle. Now slowly what happened, my dear children? It transformed. The primary oocyte remains the same. The cells also remain the same, just that you get some additional cells around it. You get some additional cells around it, and you will call this as secondary follicle. And not just that, you will now also have one more layer around it, that is known as theca layer. But what was inside remains the same only. Here also primary oocyte, here also primary oocyte. And the follicle that is being made over here is not the same. It is known as the secondary follicle. Finally, the primary oocyte is going to divide inside another follicle which is known as the tertiary follicle. So what happens? The primary oocyte is going to divide, is going to give me secondary oocyte and also the first polar body out there. Secondary oocyte is going to be diploid in nature. All the cells over here remains the same itself. You will have additional cells, that's all. So there will be cells around it, multiple cell layers. Just one very important thing happens. There will be a cavity being formed over here. This is known as the antral cavity. And not just that, one more thing happens. The theca layer which was formed over here will divide into inner theca interna and outer theca externa. Now this is known as what? The tertiary follicle. So this is the question. When does the primary oocyte transform into secondary oocyte? In which of the follicle? It will be in the tertiary follicle. Your answer for this question is C. Make sure you know about everything. Okay? How many CA students for ma'am's teaching? Oh my god. I don't think anyone is there as of now. Okay, anyways, that's so sweet of you for asking though. Now the next question that we have, the cells that secrete testicular hormones, androgens are. Now this is something very easy. If at all you get a question like this, probably you would have to thank PU board really well. 2B is the answer. Yes, 2B is the answer. That is Leydig cells. Leydig cells are the one that are going to release testicular hormones. So remember, you have the testes. Inside that you have the team lead. I always remember like this. This is team lead, testicular lobules. Inside the team leads, my dear children, you have some sub things out there which is known as seminiferous tubules that looks like this. So suppose these are two seminiferous tubules. Inside the seminiferous tubules, you have Sertoli cells for the nourishment of. So Sertoli cell is there, and these Sertoli cells provide nourishment to the spermatogonia and everything. That is one function. Now in between that, you have a space right up over here where what do you have? You have macha. You have the basic macha whose name is Leydig cell. You also call this as interstitial cell. My dear children, it is the one that releases the male sex hormones, that is androgens. The famous among them is testosterone, and I want to ask you a question. Apart from Leydig cells, what other cells are found over here in this particular region? Apart from Leydig cells, what other cells are found in the interstitial region? Important. Any idea? Any idea? Apart from Leydig cells, what other cells are also found over here? Interstitial cell, Leydig cell is the same thing, right? Yeah, 250 of them are there, that is still. I'm asking you, in this area, in the interstitial area, what other cell do you have apart from Leydig cell? This is from your NCERT. You should be knowing. What do you have? You have the immunological cells. Okay, you have the immunological cells which is going to fight against any pathogen that comes within. So always remember that also. You have some immunological cells over here. Do not discard them. This is super important. Apart from that, obviously you have some blood vessels as well. So blood vessels, immunological cells, and finally the Leydig cells would also be there. Okay? No, guys, if at all you have understood it, Just gave me a thumbs up. Those of you who still have any doubt, please ask me. Okay, do not take this doubt forward. With this, we understand that we need to read our NCERT properly, right? So within that, within the seminiferous tubule, you have circular cells, which helps nourishment, which gives nourishment to the spermatogonium and everything. In between that, you have Leydig cells over here in the interstitial space, and they release the androgens, male sex hormones. Apart from that, you have immunological cells and also some blood vessels. Do not forget it. Do not mess around it. Yeah. Yes. Okay, Nandish Reddy, hello. Okay, next question that we have is, which of the following statement is true? We solved this question before. LH surge induces menstruation, regression of corpus luteum increases progesterone, diploid egg is formed after first meiotic division, embryo differentiate from the inner cell mass of the blastocyst. Which statement is going to be true? That is the question. Sanjeevani says the answer for this question is 3D. What about others? What are my other little chipmunks out there? Shalom says it is D. Shalom also says it is D. D is what? Embryo differentiate from the inner cell mass of the blastocyst. You are being asked what is true, okay? I hope you guys remember it. Nitin is also saying D is the answer for this particular question. Okay. I think since most of you guys are saying D, we would have to lock option number D itself. Listen, LH surge induces menstruation. That would be a wrong statement, because when LH is surging up, it is inducing ovulation. The follicles which were mature until now, they burst open and finally release the secondary oocyte. That is known as ovulation, not menstruation. This is wrong. Now, regress of corpus luteum increases progesterone. Listen to me. Corpus luteum is something that is going to maintain the progesterone. So if corpus luteum is up, progesterone is also going to be up. Now you are saying corpus luteum is less, so it will increase the progesterone. That will be wrong statement, right? Diploid egg is formed after second meiotic division. There is nothing that is known as diploid egg. Okay, egg is also always going to be haploid. A normal cell is diploid in nature. That is, a primary oocyte can be diploid in nature. When it divides, when it undergo meiosis, it divides, it now forms only haploid cells. And that is why your egg is also going to be haploid. Now, embryo differentiate from the inner cell mass of the blastocyst. Blastocyst is what, Baba? Blastocyst is a, what do you call, is a stage of the embryo where you have outer layer of cells which is known as trophoblast. Okay, and inner layer of cells which is known as the inner cell mass. Inner cell mass. So the outer layer of cells which are there, they basically help in the formation of placenta, and the inner cell mass of this particular layer actually forms the embryo later on. So even though we call the whole thing as an embryo, but please understand the outer layer is there for formation of placenta, and the inner one is the actual one that forms the embryo later one, which is why we say that the answer for this question is going to be D. Haploid egg is formed. Exactly, Tanu. Cool. So your answer is D, and let's go with the new question that is out there. Try it out. Scrotum helps in maintaining the temperature of testis, which is lower than the normal body temperature. How much degree lower than the normal body temperature? Whenever you have got multiple things like that, solve one by one. How much degree lower than the normal body temperature? Scrotum helps in maintaining temperature of the testis, which is lower than the normal body temperature. Each of the testes has dash compartments and measures dash centimeter in length. Sanjeevani says the answer for this question is D. Shalom? Oh my God, we are solving for fourth question. Mansi is there. Hello Mansi. Hello Arib. Okay, so both of you, okay. Answer would be what? Fourth answer would be A, B, C, D. Okay, Jia says the answer for this question is D. And Shalom says B. Tanu also says D. And answer is 4. Answer is 4th, as in you mean this? Okay, let me see. D is what? 2 to 2.5 degrees Celsius lesser than the normal body temperature. Exactly, it is 2 to 2.5 degrees Celsius. That is this. Okay, now you have two options like that. You have this option like that, and you have this option like that. Let's go ahead with the next thing. How many compartments are there in the testis? 250 of them. Again, this is going to be the same in both of them. By the way, we have eliminated both these choices out there. Now, it measures dash centimeter in length. What is the length of the testis? What is the length of the testis? This is super important. So some of you said that D is the answer. Length is 2 to 3 centimeter. Baba, it's not 2 to 3 centimeter. Listen, if this is a testis, the width is going to be 2 to 3 centimeter. The length is 4 to 5 centimeter. 2, 3, 4, 5. Remember like that. Over here, your answer for this question is going to be B. It is going to be B. Okay, it's not D. It is going to be B. So who gave me the answer as... I think Shalam gave me the answer and Malsi also gave me the answer as B. And yes, that's all. Tanu also gave me the answer as B. Mohan, hello dear. B. Anju also says now it's B. So please listen to me. Do not do this mistake again. And be sure that you are very pro at all these small details out there. Okay, I'm telling you, when finally you're done with biology and when you're doing the revision, just take your question bank and try solving questions out there. Check if you're able to solve it or not. Okay, that's the best way to revise. Now, okay, next question that we have is, in spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is going to be affected? Come on. If in spermatogenesis, if FSH hormone is not secreted from the anterior pituitary gland, which stage is going to be affected out there? Any idea? Fifth, B is the answer. Mansi says it's B and Sanjeevani also says it is B. What about others? B means formation of spermatozoa. Now I would love to hear all of your answers. Shalam also says B. Anju Creations also says it is B. Okay, since everyone is saying B, probably I would have to look for B only. So FSH is not secreted from the anterior pituitary. What does FSH do? Suppose it is secreted. What does it do? It goes and signals the Sertoli cells. To what? To produce some factors. To produce some factors. Why do you think these factors are super important? Listen to me carefully. So for spermatogenesis to happen, my dear children, for spermatogenesis to happen very smoothly, you need a lot of testosterone. That is the male sex hormone out there, right? And there should be concentration of the male sex hormone being maintained up over here. Who makes sure that basically the testosterone which is released by the Leydig cells would be found in more amount over here? Who makes sure of that? It is the factors which are being released by the Sertoli cells. And you need to understand that yes, testosterone is needed throughout the process, but majorly during one particular process where spermatids, my dear children, are transformed into spermatozoa. So after meiotic division, all the sperms are like golu. But do we need golu sperms? No, we need elongated sperms. And that process of basically transforming the spermatids, that is basically kids, and kids, they are immature. You can remember like that. Spermatids are the immature version. We are transferring into spermatozoa. Finally, they look like this. And this process, my dear children, is known as Spermatogenesis. Spermatogenesis. So during this particular process, you need an active, very high concentration of testosterone, for which you need these factors as well. So if FSH is not secreted from the anterior pituitary, which stage is majorly going to be affected? This stage, that is formation of spermatozoa, is the one that is majorly going to be affected. Always remember this. Aditya, hello. Hey. And so answer for this question is B. I hope everyone got it. So I see the other children who gave me the answer first gave me the answer correctly. Tanu also gave me the answer. Mohan, my dear child, please can you check with it? Is it fine now? Did you understand this question? Or if you have any confusion, please ask me. Gia also says the answer for this question is B. So I hope this is clear. If this is clear, just give me a quick thumbs up. Anju ma'am, molecular basis, I can't understand. Please explain. Molecular basis, listen, I have done a master stroke video. If at all you can, please watch it. You just have to type master stroke and whatever the name of the chapter is there. That is definitely going to help you out. It's a one-shot video. If you watch that, it will be all clear. Okay, Sanjeevani, Shalam, thank you for giving me thumbs up. We'll go ahead probably. The next question out there. To produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required over here? Thank you, Mansi. Ma'am, organism and population need explanation. We'll solve questions, okay? Anju, so previous year there was a typical list for HOTS question out there that they gave in the blueprint itself. This year they have not given. So I'm expecting from, one from sexual reproduction and flowering plants, one from molecular basis of inheritance and principles of inheritance. These are the chapters that can give you HOTS question. Okay. Okay, Jia says the answer for this question is C. Hello Pooja. And okay, Mansi says it is B. Nagaraj is there. Hello Nagaraj. Says it is B. Tanu says B. Anju Creations. Okay, ma'am. And mostly I see the options that you are giving me is B. B is what? B is 500 spermatogonia and 400 oogonia. Let me see. So to produce 200,000 sperm and 400 ova, how many spermatogonia required? One spermatogonia, if at all we think about it, basically they give us 4 sperms at the end. How? Okay, you got the answer, but still they give 4 sperms at the end. So I am saying 2,000 sperms are already being formed. If I do divide 2,000 divided by 4, what do I get? I get 500. So basically 500 spermatogonia were there. They divided, each of them gave 4 sperms, and that is how I got 2,000 sperms at the end. So 500 spermatogonia is correct. What about the oogonia? Listen, when I say that one oogonia is going to divide, you need to understand that one oogonia at the end will only give 1 ova. Why? Because the division over here, it's like that. This guy divides, the primary oocyte divides into 2. It is unequal division, one polar body and only one normal cell is being formed. This cell is not going to undergo division, which is why again, when it divides, again the next polar body and one cell. So basically I can just count one cell is being formed at the end. So when I say 400 ova are being produced, that means 400 oogonia will also be there. It's the opposite over here. So your answer for this question becomes B. I hope this is clear. Yes. Monar, perfect dear. Shalom and Anju Creations, everyone are there. Perfect. Now, let's go ahead with the next question out there. Question number 7. Which among the following has 23 chromosomes? 23 chromosomes. Which among the following has 23 chromosomes? Come on. Spermatogonia, zygote, secondary oocyte and oogonia. 23 chromosomes means basically talking about haploid content, okay? Mansi says the answer for this question is C. Tanu says it is D. Baswaraj is there. Hello Baswaraj says it is D. So A, B, C, D, just A is left for me. Please give me A option also. Everyone. Nagaraj says C. So C is something that I think is getting the maximum votes. Sanjeevani also says C. Jia says D. Okay. Anju also says D, which is why probably I have to look for the option C or D. Have you thought really well before typing the answer? This is super important, okay? Which of the following has 23 chromosomes? Spermatogonia, Baba, it's a normal cell. It is the one that will be later helping in spermatogenesis. It is diploid in nature. Zygote, definitely male and female gamete comes together, diploid in nature. Secondary oocyte, which is there. What happens? Primary oocyte, 2n. It undergoes meiosis, and the end of meiosis 1, you get 2 cells, and obviously after the end of meiosis 1, it becomes half. So secondary oocyte is definitely going to be haploid, that is 23 number of chromosomes are going to be there. Now oogonia, what about oogonia? Oogonia is basically just above this, that is also going to be diploid only. So your answer for this question is C. Please make sure you go through it, do not get it wrong, read the questions carefully. Okay, so everyone who got wrong answers, I hope you have corrected it, and please understand they can give you the questions in different format. They can say that if 46 number of they can give 96 is the normal number of chromosomes in a normal cell. Okay, then which cells will be having a 43 number of chromosomes? Not 43, sorry. Okay, so the question can be, suppose I am saying 86 number of cells are found in the, 86 number of chromosomes are found in a normal cell, and in that case 43 number of chromosomes can be found in which cells? So please know 86 means over here it is diploid in nature. Half of it is what? 43. So basically half of it will be only found in the gametic cells, and in the gametic cells you have stages. Primary oocyte, primary spermatocyte, you will not find it. From secondary stage only it will be there. So please know that. Okay, very important. Understood? Okay, perfect. So let's go ahead with the next question. Question number 8. Identify the wrong pair with respect to menstrual cycle. Come on. This question I want everyone to read out carefully and then only answer this. Okay, super important. Identify the wrong pair with respect to menstrual cycle. Formation of corpus luteum 15 to 28 day. Secretion of progesterone 15 to 28 day. Regeneration of endometrium 5th to 13 day. Disintegration of endometrium 5th to 13 day. Super important question. Try to find it. Nagara says the answer for this question is D. Hello Sunil. Good morning, by the way, and it's D. Sanjeevani says it is D. Anju also says D. Baswaraj D. Oh my God, everyone is saying D. Nagaraj, Anju, and okay, you guys are super sure about D. D is what? Disintegration of endometrium 5th to 13th day. Is that so? Listen to me very carefully. Oh wait, where is the line? Yeah, this is the line. This is the 1st to 5th day. This is going to be the 28th day, suppose. Half of 28 is 14th. On 14th day, what do you have? You have ovulation. Done? Done. Okay. Now, menstrual phase is this. That means during this phase, disintegration of endometrium has to happen. Disintegration of endometrium from 5th to 13th day. Wrong, no. It should happen from 1st to 5th day. From 5th to 13th day, Baba, it is going to regenerate. So this particular statement is going to be wrong, and that is what you have been asked. Which is the wrong pair? Why are others correct? Formation of corpus luteum 5th to 28th day. Listen to me. What is corpus luteum? Once your Graafian follicle ruptures and releases the secondary oocyte, during ovulation, whatever is left behind is known as corpus luteum, and that will be there. Okay, that will be there from the 15th to the 28th day. It will slowly regress, but yes, it is going to be there. So this is correct. Secretion of progesterone from 5th to 28th day. How? So basically this, what do you call, corpus luteum, which is there, it is the only one which is going to secrete progesterone. So which is why that is also correct. So whenever corpus luteum will be there from the 5th to 28th day, 15th to 28th day, progesterone will also be released from the same days only. Correct. Regeneration of endometrium from 5th to 13th day, yes. So it went down and then it is slowly regenerating from 5th to 13th day, which is why this is also correct. What is your answer? Your answer for this question is D. I hope everyone, I think no one got it wrong, but if at all you got it wrong, or if at all you had a confusion, I hope that's clear. If yes, just give me a quick thumbs up, everyone. 3 to 5 days disintegration, exactly. Everyone who got it crystal clear, just give me a thumbs up. Thank you, Shalam. Thank you, Jia. Thank you, Sanjeevani, Mansi, and Anju. Perfect. Now, question number 9. The secondary oocyte after ovulation is covered by a non-cellular layer of secondary oocyte, which is being released, right? It is covered by a non-cellular layer of. So, I hope you guys know that primary oocyte, secondary oocyte, one meiosis happens. Second meiosis has not completed until fertilization reaches. Okay, until that time reaches out there. Sanjeevani says the answer for this question is C. Baswaraj C. Sunil also. But Sunil has a different answer. Mansi says C. Nagaraj says C. Anju says C. YouTuber is there. Hello, YouTube star. Princess Aline. Hello, Princess Aline. And yes. So most of you are saying C, and I hope you are not thinking that others are saying C, which is why my answer should also be C. Don't do like that. You will mess around the paper, okay? So the answer is actually zona pellucida. So listen to me. The secondary oocyte, which is when it is there inside the Graafian follicle, it is the one that makes its own layer around, and that layer is known as zona pellucida. And zona pellucida. And some unique feature about zona pellucida is that it does not have any cells. It is non-cellular. Always remember, it is a cellular layer. Around it, you have some more cells out there. You have some more cells, and that is known as corona radiata. But this is not something that the secondary oocyte makes. It is the one that is contributed from the granulocyte cells of the Graafian follicle. So inside the follicle, all of this is happening. This guy makes its own layer, that is zona pellucida. Please remember that, and that's all. Okay, and zona pellucida will go away only after fertilization. Okay, only after fertilization, zona pellucida is going to go away. Until then, it is going to be there. Please remember it. So this guy, secondary oocyte, will further divide, undergo meiosis to form ovum, as well as the second polar body when the sperm is going to come very near. And after this has taken place, and after the ovum and the sperm have fused together, later on, slowly the zona pellucida layer is going to disintegrate or dissolute completely. In between that, just to allow the entry of sperm, it slowly widens up a bit. There's a space in bit, but does not mean that it gets destroyed. It gets destroyed only after fertilization. I'm telling you again, please remember it. Okay, ma'am, I like your class. Thank you, Baswaraj. That's so sweet of you, dear. Okay, next question. Question number 10. During pregnancy, the fetus develops limbs and digits when? The fetus is going to develop limbs and digits when? Come on. During pregnancy, the fetus develops limbs and digits. Hello, RMS. Inchara. Hello, dear. Okay. Good morning, good morning. Okay. 10B, Shalam, Sunil, Anju, and Sanjeevani, and Nagaraj. I hope I'm not missing out on anything. Okay, Anju, Nagaraj, Inchara, B is what? B is after 8 weeks. After 8 weeks, are you sure about that? Are you sure about that? Think about it. Listen to me. First month, second month, third month, fourth month skip, fifth month and sixth month. This is how it goes, yeah? 1 month has how many weeks, Baba? 1 month has 4 weeks. So I'll say this is 4 weeks. 2 weeks means 2 into 4, 8 weeks. Okay, this is 8 weeks. 3 into 4, 12 weeks. 5 into 4, 20 weeks. 6 into 4, 24 weeks. Okay, please remember it. This is a week by week data. Now, month by month data, if at all we go ahead with that. Okay, if at all we go ahead with that, by the end of one particular month, the first month, my dear children, dhak, 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 dhak is going to be seen. That means your heart is being formed right over here. So when someone dies, that's the first thing that you check. When someone is born, that's also first thing that you check out there, right? Now the next thing that you have is second month. Second month is the time when you have all the limbs and digits being formed. So you guys are correct with your answer. Okay, the limbs are this and the digits are these. These things are going to be formed by the end of second month. Now, by the end of third month, what is going to happen? Oh, limbs and digits are being formed. The extra organs which are there, all the organ system which is there, it is going to be formed. The organ system, it is going to be formed. Apart from that, external genital organ, that means for a female, it will be what do you call the mons pubis and everything out there. Similarly for males, the penis is going to be formed. So external genital organs are going to be formed by this particular month. So by 3 months we say that, okay, by 3 months, if at all you have seen, the ladies who are pregnant, they reveal their pregnancy by the end of 3 months. Right? This is a very crucial time period that is there. By the end of that, almost so many things are being formed out there. Okay. Now by the 5th month, baby says that, God, I want to show some style. I need some hair, macha. So please give me some hair. So God said that, okay, you want some hair, I'll give you some hair. So there will be hair on the head. Hair will be there on the head, but the baby says, I want to move around, I want to dance also, just give me some movement also. So there will be some movement. The first movement, always remember, first movement, fur over here. First movement, 5th month, always remember, and hair will be on the head. Now God said that you wanted, macha, you wanted hair, no? Take hair on your body also. So in the 6th month, they will be having small hair on the body, and apart from that, the eyelids which are there, they are going to get separated. They will be having eyelashes and everything. So everything hairy happens over here. Eyelashes and also the eyelids are going to get separated. And this is how you have to learn, and make sure you learn like this. You will not forget it. You can get any question on the basis of that. Do not mess it around. Okay, so your answer for this question is after 8 weeks, because 8 weeks means 2 months. Perfect. All clear? Yes, perfect. Next question that we have is from the previous year model question paper. Neural tube formation, limbs and digits, organ, hairpins, eyelids separate, fully developed fetus. Exactly. So many things that are happening up there. Great. Perfect. Now the next question that is there, statement number 1, the process of release of sperms from the seminiferous tubule is called spermatogenesis. Statement number 2, the spermatids are transformed into spermatozoa by the process of spermiogenesis. What do you think is the answer for this particular question? Shalom says 11A, Anju says A, Sanjeevani says D, Shalom says A, Nagraj says D. So 2 people for D is what I am saying. Mansi also says D only. Rest everyone is mostly saying A. Shalom now says D. Okay, Nagraj also D. So I think there is a typo, and yes, Inchara has a different answer. Let me see. So process of release of sperms from the seminiferous tubules is not known as spermatogenesis. Spermatogenesis means formation of sperms. The release of sperm is spermiation. Aviation, you know, aviation course is there. Similarly, you have to remember spermiation. Okay, so here this is wrong. The answer should be spermiation. Now, what do we have next? Statement number 2. The spermatids are transformed into spermatozoa by the process of spermiogenesis. The spermatids which are there, they are transformed into spermatozoa. I told you, immature kids, that is spermatids, right? They transform into spermatozoa, that is long sperms, by the process of spermiogenesis. That is actually correct. So this particular statement will be true. So statement 1 is false and statement 2 is correct. So your answer for this question is going to be D. Okay? It happens, do not be sad. And I'm really glad that you guys are trying. You know what? Please do not remove your messages and do not retract your messages. whom are you showing? Whom are you not trying to show? Or basically, whom are you hiding from? It's you only who know your answers. Remember, neither anyone, for example, I am not going to remember who got your answers wrong. Neither Anju is going to remember who got your answers wrong after solving so many questions out there. Do not retract your message. Keep it there itself. Work on it. That is how we have to face our mistakes. That is how we learn better. Okay, always remember that. Yes. And next question that we have over here is this. Come on. Choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. For the formation and dissolution of the labeled part in the given diagram. Choose the correct option from the table given below for the formation and dissolution of the table part in the correct diagram. So this particular diagram is given. Zona pellucida is given over here, and you have been asked what is it formed by and when it is going to be dissolved. What do you think is the answer for this? Mansi says the answer is C. Okay. Sunil says the answer is D. I get two options. What about others? Sanjeevani says it is D. Okay. Sunil now has changed. Okay. And Shalam says it is D. Inchara says D. Jaya also says it is D, 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 D. So what is D? Secondary oocyte and after fertilization? Listen to me. It is formed by secondary oocyte. We just discussed about it. Zona pellucida is formed by secondary oocyte. Dissolved after fertilization only. I told about that also. So some people might think for the sperm to come in, you have to dissolve the zona pellucida. It is not dissolved entirely, na baba. Only at one place or two places it will be dissolved, and that's how the sperm gets in. And finally, after the sperm gets in, once sperm gets in, it will become very thick also, the zona pellucida. Later on, after fertilization, this is going to be dissolved completely. So you should know the answer for this question. It is going to be D. Okay, clear? Hello Bhuvan, I welcome you all to our class. I hope this is clear. If it's clear, just give me a quick thumbs up, everyone out there. Perfect. Thank you, Mansi. And my dear children, I hope you have shared. Hi Gauri, hi Gauri. And thank you everyone, Inchara, Sanjeevani and Bhuvan and Anju Creation. And okay, I think we are going ahead with the next chapter. But before I go ahead with that, everyone who is preparing for KCET, my dear children, a quick update for you guys. There is KC30 online crash course, and it is going to begin from 21st of March. So if at all you are someone who is interested in KCET, please take up this course. The price offer that we have as of now is it is available for ₹2499, right? And now if you prefer to study offline, not online but offline, there is Lakshya KCET offline crash course. There's a flight 3,000 discount going on. Make sure you check it out. And there is ABHYAS KCET test series. If at all you do not want any of the crash course, the test series is just available for ₹999. And trust me on this, so many children got the best benefit of it the previous year. There were so many rank holders. So if you're serious about KCET and you seriously think that I need a guidance, you can go for it. If you think that I can do it by myself, you do not need to take anything out there. Okay? Yes. And now we go ahead with the chapter reproductive health. And now, before I go ahead, I see the number is counting to be less, and I'm pretty surprised by this number. Why? Because I remember, as I said, in the WhatsApp poll, there were almost 359 children who voted for 13th morning live. And where are my children over here? I do not see those. So make sure, if you have your friends out there who voted, or probably you know that they needed MCQ questions and everything like that, ask them to join the live as soon as possible. Okay? Yes. This number is kind of demotivating at this time when you have examination, right? Yes. Perfect. Now, reproductive health, we go ahead with the first question that is there in the MQP. Oral pills as contraceptive effectively function by inhibiting what? What does it inhibit? Oral pills which are there, what does it inhibiting? Malleshwar, ma'am, all are preparing for maths. I definitely agree. But yes, so with the votes, that's exactly why I did a voting out there. I thought I should be taking it on 12th. But since most of them voted for 13th, I thought let's do it on 13th then. So I think 12th would have been a better option for children who actually wanted to see it. Yes. But you guys are there and I'm super proud of you. Okay, so ovulation. Yeah, my friends are CSS. It's okay. So sweet of you. It's okay, guys. Let's continue, okay? So ovulation is what everyone is saying. And since you guys are saying ovulation, let's go ahead with it. Ovulation is the answer. So oral pills, Baba, what do they contain? They contain progesterone or estrogen, or basically both of them together. And these two hormones are super important for the process of ovulation to happen, for the process of implantation to happen, for everything good in our body. But when you give them from external source, the level of it is going to get imbalanced, which is why the normal functions which were happening, for example ovulation, it is not going to happen. The normal function of basically thickening the endometrium, it is not going to happen, rather ulta will be happening. Thin endometrium is going to be there. Do you think the embryo will go and attach to that thin endometrium? Obviously no. Obviously no. So please make sure you understand that oral pills or any other hormone related thing messes up with the ovulation, implantation, and also they form a mucus plug towards the cervix such that the sperm cannot go in. Yeah, so yes, so your answer for this question is going to be ovulation. Next question that you have, which of the following is not a copper releasing IUD? What do you think is the answer for this? Which of them is not a copper releasing IUD? Anju, ma'am, can you please do a live on 14th? Please, definitely I'll be doing that. So 14th, after your mathematics examination, at least some of your mathematics examination, I'll be coming live with complete biology. It will be a long live. Make sure you're prepared for that. And yes. A. Okay. So Sanjini says it is A. Malleshwari is there. Hello, dear. Says it's A. Mansi also says it's A. Bhuvan says A. Gauri Patil. LNG 20. Yes. Okay. Bhuvan also says A. Ankita A. Okay. Jia A. Perfect. So the answer is LNG 20, and Chalam says it is hormone releasing. Yes, it is hormone releasing. It is not copper releasing IUD. So in copper releasing IUD, you just have three of them, CUT, QT. Okay, it is a T-shaped thing on which your copper is wrapped. CU7, it's a 7-shaped thing in which your copper is wrapped. And multiload 375, it looks something like this. Again, over here, the copper will be there in high amount out there. And all of them are copper releasing IUDs. What does the copper do? Copper says, hey sperm, you cannot move around anywhere. So basically, it holds the tail of that. You just remember it like that. It's for fun, but actually it messes around the motility of the sperm and does not allow the sperm to fertilize with the egg. So it reduces the fertilizing capacity and also the motility of the sperm. LNG20, levonorgestrel, it is hormone releasing IUD, as you guys told me, which is why this one is the odd one out there. Yes? Yes. Now, next question that we have is this. Statement number 1, gift is an in vitro fertilization technique. Statement number 2, in gift, gametes are fertilized outside the female body. So first 3 questions are from MQP. I believe that most of you should be able to solve that. Yeah, Sanjini says the answer for this question is B. Oh my God, Sanjini is full josh. Okay, Malleshwari also says B, Bhuvan also says B, Gauri C, Mansi B, Gauri B, Mansi B, and G also B, Ankit also, Shalam also B. Okay, so both statement 1 and 2 is correct. Is what that you're saying? Is what that you are saying? Is it B? Guys, I'll kill you. Think. Come on. Think. Everyone, think. Listen, we cannot afford to do mistakes over here today. Yes, ma'am, it's correct. No, it is not correct. A is not correct. Sorry, B is not correct. That's what I meant to say. Oh, yeah, sorry. B is correct. Sorry, Shaktiman. That was my problem. I read A option as B option. Sorry. Sorry for giving you a small heart attack out there. That's my problem. Gift is an in vitro fertilization technique. It is wrong. Why? Because it is in vivo fertilization technique. In vivo fertilization technique. Now, in gift, gametes are fertilized outside the female body. Gametes are they fertilized outside the female body? No, Baba. Gametes are put inside the female body. Fertilization happens within only. So both the statements are going to be incorrect. So I read this option for B and I was saying, I do not know. My mind was out. Heart attack, ma'am. Yes, I know. So I hope everyone who was feeling is not sleepy anymore. Anyways, so the answer for this question is going to be B, and yes. So I felt bad more for the children who attended my model question paper live, and now when I said it's wrong. So yes. Okay, next question. Out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. Read the question carefully. This is from the previous year model question paper, okay? Mini heart attack, I know. Okay. So, out of the options given below, choose the correct stage for transfer to the fallopian tube for successful IVF results. What do you think is the answer for this question? Transfer to the fallopian tube. Sanjeevani says A. Maleshwari says B. Okay, now this time I have to look at the options. A, B, okay. What about others? A. Mansi says A. Shalam also says A. D also says A. A has maximum votes, which is why we shall look into A. What is A? A is embryo up to 8 blastomere stage. Is that so? Listen to me. You are transferring the embryo in IVF, you're transferring the embryo in the fallopian tube or anything in the fallopian tube. It has to be either a zygote. Zygote is having only one cell, or it can be an embryo. If it is an embryo, the embryo need to have not more than 8 blastomeres. If it is more than 8 blastomeres, we cannot put it inside the fallopian tube, which is why your answer for this question is going to be A, Baba. Okay, since fallopian tube is mentioned. And what do you call this process as? Come on, what do you call this process as, where you put the, this particular gamete inside? When you put the zygote or when you put the embryo up to 8 blastomeres inside the fallopian tube, what do you call this process as? Zift, exactly, Maleshwari. Zygote intrafallopian transfer. So it can be a zygote or also an embryo up to 8 blastomeres. Zift, exactly, Sanjeevani. Shalam, Zift, perfect, perfect. Mansi, Jia, everyone correct. Now, question number 5. Let's solve this. Reasons for human population explosions are given below. You have to select the correct answer from the options given below. Come on. What do you think is the correct answer for this? Reasons for human population explosions are given below, and you have to choose the answer statements, correct answer statements from the options. These are the options are there. I'll move aside. You can go ahead and try to solve it. Five C is the answer. 5C. Shalam also says C. Maleshwari also C. C is what? C is this. Okay. I have to look into the answer then. Rapid decline in maternal mortality rate is a reason for population explosion. Yes. When mothers are dying less, obviously the population is increasing. Okay. Perfect. This is correct. Rapid decline in infant mortality rate. If infants are dying less, obviously population is increasing. Yes. Rapid increase in death rate. If someone is dying, how do you think population growth rate is going to increase? That is wrong, right? Wrong statement. Now, increase in the number of people in the reproducible age. So, number of people in the reproducible age, if they are more, obviously in future they will be reproducing. So, population is going to increase only. So, obviously this is also reason for population explosion. So, can I say 1, 2 and 4 is the answer? And in that case, your answer for this question is going to be C. I hope no one gave any other answer out there. Okay, so the answer over here is going to be C. Now going ahead with the next question out there. Question number 6. Which of the following sexually transmitted diseases is not completely curable? It is not completely curable. What do you think is the answer for this particular question? Maleshwari says C. Okay, Sanjeevani says C. And Mansi C. Shalam also C. What about others? Other children who are watching this video, come on. Come on, machas. Listen, I am not here to judge you guys by any chance if at all you think like that. I'll be more than happy if at all you just attempt the answers, okay? Anyone out there with any separate answers for this question? G also says the answer for this question is C. C is what? Genital herpes. Why do you think it is not completely curable? Gonorrhea, genital warts and chlamydiasis is what you say is completely curable? Is that so? Is that what you're going to say? The answer is correct. It is not completely curable. Remember, there are 3 H which are not completely curable. HIV, AIDS, hepatitis B, and finally herpes. The actual name of this herpes is genital herpes. There are different forms of herpes. This is going to be genital herpes. These 3 things are not going to be completely curable, which is why your answer for this question becomes C. It becomes C. I hope this is clear. HIV and hepatitis B. Exactly. Okay? Yes. Perfecto. Let's go ahead with the next question then. An example for non-medicated IUD is non-medicated IUD. That means nothing. It is an intrauterine device. It is put inside the uterus, but it is not medicated. It is not going to release anything out there inside the uterus. What is the example for that? That is the question. 7B. Mansi, Malleshwari, Sanjeevani, Shalam and Jigar. Hello Jigar. Okay, what about others? Any other options for me? G also says it is B. B is what? Lipase loop. Then what does this non-medicated IUD do inside our body such that it prevents conception? Listen to me carefully. Suppose this is the uterus and this is the lipase loop. It looks like a loop sort of thing and it is fitting our uterus really well, and it acts like a foreign thing. For example, whenever our body encounters something foreign, what is going to happen? Basically, it is going to be very active. Its immunity level goes very high and it is ready with so many cells to attack. Now suppose when this guy is up there, the lipase loop is up there put inside the uterus, and suppose a normal insemination happens, the sperm comes inside. All the cells which are over here lined up, okay, all the cells, immunological cells which are lined up, they are going to kill the sperms, and that is how they are going to die. So basically, I'll say that they are responsible for phagocytosis of sperms. Phagocytosis. They eat up the sperm. Who eat up the sperm? Not the lipase loop, but the cells around it. Okay? They are going to eat up the sperms. So your answer for this question is going to be B. Now with the next question that we have, question number 8, the assisted reproductive technologies in which a sperm is directly injected into the ovum is. The sperm, when it is directly injected inside the ovum, what do you call this? Malleshwari, I'm assuming this is going to end by 12:30. That's what I promised. Okay, if it ends before, I'll be more than happy, but beyond 12:30, we'll not take it up. Okay? 8A, because there are so many questions, right? It might take some time. That's the reason I've said 12:30. Jigar says 8A. Okay, what about Shalam? Shalam also says A. Malleshwari also says A. Mansi, Jia also says A. A is what? ICSI. ICSI stands for what? Intracytoplasmic sperm injection. Intracytoplasm. Inside cytoplasm of what? Of the egg. What are you injecting? Sperm injection. Sperm is something that you're injecting. Why you are doing it? Such that fertilization can happen in vitro in the laboratory condition. So the reproductive technology in which sperm is directly injected into the ovum would be ICSI. Your answer is A. Next question. Coitus must be avoided from day 10 to day 17 of the menstrual cycle. The reason is what? Why do you think that the coitus should be avoided? Ma'am, I was absent for my health issues. Can I write the second examination? Dear, we will be making a separate video on it. So we have to still talk with the Dean of Deeksha to just have a clarity on what is being put by PU board. Okay, there might be certain rules out there, so I'll just check it out and surely would make a video really soon on it and would be publishing it. I hope that would be answering your question, right? Yes, Sanjeevani, the answer for this question is B. Most probably you should be able to write there. Okay, Sanjeevani says the answer is B. Why not? Is also saying, nice name, says B. Okay, Jigar says C. Mazi says B. Arif says B. Shalam also says B. What is B? Ovum may be available for fertilization. That is why you should be avoiding coitus from day 10 to day 17 of the menstrual cycle. Is that so? Yes. So listen to me carefully. Suppose a woman who is there, I told you the cycle is from 1st to 28th day usually, and the middle of that is 14th. So we expect that on 14th day what is going to happen? Basically ovulation is going to happen. Ovulation is going to happen. Now it's not like correct on the 14th day ovulation can happen. It can happen few days before, it can happen few days after also. We never know. So we say from 10 to 17 days. From 10 to 17 days, this is what? This is a fertile window. Fertile window where the ovum, my dear children, can be released. Okay, where the ovum can be released. Now suppose the sperm which are there, the sperms, my dear children, which are put inside the female body, they can remain viable inside the female body for 5 days, 5 whole days. So suppose it was really, what do you call it? The sperm were released inside on 11th, and within a few days ovum was also released. Obviously fertilization would be happening, right? So which is why we say that ovum may be available for fertilization. That is exact reason that you should be following periodic abstinence. This process known as periodic abstinence, where during a certain period you are absent, you are not doing any sort of sexual activity. I hope that answers yes. So for this it is B. Good. For everyone who are giving me the correct answers, awesome. For everyone who are trying, awesome. Okay, next question that is there. Condoms have additional benefit as compared to the other contraceptive because what benefits do you think condoms would be having? Condoms would be having some additional benefits as compared to the other contraceptive because of what? Because of what? It is made of rubber latex, provides privacy to her user, protects against STIs, or it prevents implantation. What do you think is the answer for this particular question? Ma'am, in open hair, you look more beautiful. Thank you. Gia says D, and Gia, I hope you are answering this question. Yes? Okay, Sanjeevani says the, I do not know. Mansi says the answer for 10 is C. Sanjeevani says it is C. Govardhana, hello dear, says it's C. Jigar, okay, and everyone. Shalom also, Inchara also, Arib also, everyone says it is C. Protects against STIs. Okay, so condoms have some additional benefits in terms of what? In terms of providing protection against STIs, that is absolutely true. Why true? But listen to me carefully, you should know the reason. So the penis which is there, it is being covered by the latex, right? Rubber latex. And suppose it has been inserted in the female genital tract. Do you think any of the fluid exchange is happening out there? No fluids are being exchanged over here, which is exactly why it can prevent any of the STIs. So STIs usually transmit through what? The genital fluids, or it can be through the blood or things like that. Some of them transfer also through the blood, some only through genital fluids are there. So anyway, condom is something that does not allow it to happen. Okay, so physical barrier that does not allow it to happen. So yes, your answer for this question is going to be C. Okay, typo. It's okay, dear. Perfect. Yes. Let's go ahead then. Next question, question number 11. There is a statutory ban on amniocentesis as. Why do you think there is a ban on amniocentesis? Any idea? Any idea why there is a ban on amniocentesis? 11B. Jigar also says B. It can be used for determining the sex of the inborn. Is that so? Govardhana also says it is B. It reveals the chromosomal anomalies. That is why you put a ban? No. If at all it is doing something good, why will you put a ban? It can be used to reverse the sex of the fetus. It can be used for correcting the genetic disorders of the fetus. These are all the good things. Listen to me very carefully. Amniocentesis was being done for understanding the genetic abnormalities a person could have in future. But then, since anyway the genetics, the chromosome, everything was easily being gotten, people started identifying the chromosome, sex chromosome, people started identifying the sex of the child, and later used to do female feticide. They used to kill the females out there. That is why there was a ban on amniocentesis. So please know that it is actually used for finding what? Genetic abnormalities. Example of genetic abnormalities, you have sickle cell anemia, Down syndrome and things like that. Remember, jaundice is not a genetic abnormality. You cannot find jaundice with the help of this. So the only answer for this question is going to be B. Okay? Perfect. I see a new person out there. Hello, Varsha. Okay. Now the next question. Select the option with correct identification of the given contraceptive measures and the hormone essentially present in it. So over here you have a question. You have to solve it. What is this picture about? What is this picture about? And the question is, you don't just have to say what exactly is this. You have to also say what are the hormones which are going to be released by these guys. 12D. Okay, first of all, Sanjeevani says 12C, Mansi says D, Deepthi says C, Jigar also says C. Hello, Deepthi. Arib says D, and Deepthi says implants. Exactly. Mansi says C, Shalom also says C. What is C? Subcutaneous implant. Is this a subcutaneous implant? Subcutaneous means a layer, okay, beneath that particular layer, we are going to put what? The implant. The implant is slowly going to release some hormones. What hormones? It can be either progesterone or probably a combination of progesterone, estrogen as well. These hormones are being released. Why? It does the same thing what oral pill does. What does it do? Prevents ovulation, prevents implantation, increases the cervical mucus, all of this thing. Which is why your answer for this question is going to be C. It is going to be C. I hope it's clear. Yes. Next question. Question number 13. Children, I hope that's clear. If it's clear, can you just give me a quick thumbs up? That will be a great thing to do. Thank you, Shalom. Okay. Next thing. Okay. A lady is 8 weeks pregnant. That means 8 weeks means how many months? 2 months. Have this in your head. Always have this in your head. Okay, 2 months pregnant. Various tests done confirm that if her child is born, it would suffer from a serious illness. What is the best suggested option in this particular case? She is just 2 months pregnant, and it makes us understand that the baby has a serious illness. What is something that is being suggested? Amniocentesis is to be performed with the help of, so most of the children get confused over here and here. Okay, here and here. Please understand, with the help of amniocentesis, probably you would have confirmed that yes, they have a genetic disorder. Now, the next step that you have to do is probably MTP is the only thing that is left. MTP, medical termination of pregnancy, has to be done, and it has to be done before how many weeks? Before 12 weeks, if at all possible. That would be the safest if it is done within 3 months itself. And you can go up to 6 months also, that is 24 weeks also, but it is not considered to be safe at all. So up to 24 weeks it can be performed. The safety period is only up to 12 weeks, that is 3 months. Anyways, so your answer for this question is going to be C. Okay. Heteroviratians. Oh, yes. So yes, Varsha and Jigar, you guys were correct, saying 12 months. Sorry, 12 weeks. Okay, let's go. Ayo, why have I marked this? Just a second, okay? This is the answer. Okay, next question. Which of the following contraceptive devices is having a common mechanism of action? Which of them have a common mechanism of action? This is something that can be little, confusing. Make sure you solve it. You check and then only you solve it. Walls and condoms, CUT and multilode, progesta, cert and cervical caps, malady and lipase loop. Which of the following have the same contraceptive following devices is having a common mechanism of action? That is the question. Think of it very, very clearly and then only go ahead with answering this question. B, I think. Jigar says it is B. Okay, Sanjeevani has a slight confusion. Okay, sorry. Sanjeevani has a slight confusion. Let's see, it's A or not. Think. All of you think. I really want you guys to think. Varsha says B. Shalam says B. Ganesh also saying I think it's B. Inchara says B. Jia says B. Mansi says A. Okay. So children, if at all you think, walls and condoms. Now, condom can be of different types. It can be for the male, in which it covers the entire male reproductive external genitalia. Valves which are there, they are usually cervical warts, which are put in the cervix. Now, both of them are physical barriers, I do agree, but the working, if at all you see, one provides complete protection, one does not. So I'll say it is slightly different in some ways, right? Yes. CUT and multilode, both of them are copper releasing IUDs. They have the same function. What are they going to do? They are going to mess up with the motility of the sperm and also the fertilizing capacity of the sperm. Right? Right. So this should be the answer. Let's see the next option. Progestasert and cervical cap. Progestasert is a hormone releasing IUD and cervical cap is a physical barrier. So altogether it's a no. Malady and lipase loop. Again, this is also something. There is two different things. Malady is not even there in your textbook, so we can ignore it. What are we left with? We are left with something that is super common, and that is your answer B. Your answer is going to be B. I hope you have got the answer as to why. Okay, that is also super important to understand. If it's clear, just give me a thumbs up. If it's clear, just give me a thumbs up. So in condom, not basically all type of physical barriers work for preventing STIs, do they? No. Because why? They are differently built. So yes, perfect. Thank you, children. Let's go ahead. Now, question number 15. If a female has rudimentary ovary, okay, rudimentary ovary, that means it is not fully functional, fully not working, but can provide a suitable environment for the growth of the fetus, the art technique that can be used is, the assisted reproductive technique that can be used would be what? The female has a rudimentary ovary, okay, but can provide a suitable environment for the growth. What do you think is the art technique that would be used over here? Any idea? 15th C, Jigar says 15th C. Just a second. If at all there is someone who is seeing this option, probably they'll feel like, oh my god, just a second. This is B, this is C, this is D. Yeah, now you can find. And this is A. Okay, gift. So, yeah. So I get it, even if at all you gave me the wrong alphabet, I understand it's the problem from this end. So the answer for this question is definitely gift. Why gift? Because since rudimentary ovary is there, they cannot produce the gametes. And if gamete is not being produced, how do you think basically they can do fertilization? So you just need to provide the gamete. Fertilization is going to happen in vivo only. So your answer for this question is going to be B. I hope that's clear. Yes. Okay. Perfect. Guys, we have done 3 chapters out there. And obviously there's more to practice. Please go ahead and do that whenever you have time. I'm here to make you feel confident as that at least I have done few questions of MCQs, but please understand you need to do more. Obviously you need to do more as well. Yeah, works? Okay. Perfect. And I'm super proud of everyone who have reached until now. You guys are super awesome. Now we go with yay. So I think principles of inheritance I've kept after this. So this will be yes. Anyway. So molecular basis of inheritance. Let's go with the first question that is out there. This is from your MQP. Another chemical name for the nitrogenous base thymine is. What is the next name or the other name for thymine? Any idea? What is the other name for thymine? D. Sanjeevani says the answer for this question is D. D is what? 5-methyl uracil. Is it 5-methyl uracil? And Gauri says A. Shalom says C. Jigar says B. So you have given me A, B, C, D option, guys. The other name for actually the nitrogenous base thymine is uracil, 5-methyl uracil. Listen to me carefully. So in the RNA, you had uracil instead of thymine, right? And this uracil made the RNA little more less stable. It was making the RNA a little more less stable. So obviously, when you now make a new genetic material, when you evolve, you have to make a better genetic material. So in uracil, at the 5th position, my dear children, you add a methyl group. And now it will be not known as uracil. You will rather call this as what? Thymine. So the answer is 5-methyl uracil, which is also known as thymine, and it looks something like this. So on the 5th position, you have a methyl group right up there. Okay, so don't get that wrong next time. I hope it's super clear for all of you. Now we go ahead with the next question, and this question I want everyone to answer correctly. At least those who solved MQPs with me, I want you guys to answer it correctly. Those who didn't solve, please make sure you read the question carefully before doing it. Okay, ma'am, Hot's question, 5-marker, will be doing it. So most of them, all the 5-marker video have done completely in a recorded format. If at all you have time, Shalom, and if you want to watch it, please go ahead and watch it. Okay. So I do not know in live videos how much ever I would be able to cover. I'm being very honest over here. I'm just telling that. So if you can, the recorded video for the 5-marker, I'll really appreciate it. It will be really good. It's there in the playlist of biology. Okay, so B. Sanjeevani says B is the answer. Gauri says B. Okay, and Shalom says C. Mansi says B. Shalom says C. Okay, so 2 options are there. A, B, C options are given to me. So listen to me carefully. If mRNA is like this and it undergoes translation, first of all, anything that undergoes transcription, replication, translation, everything, whatever is there, you have to look for the direction. Whenever an mRNA undergoes translation, it is in direction 5' to 3'. Here it is put in which direction? It is 3' to 5'. That means it is ulta. It is ulta, right? So that means reading will be starting from here to here rather than here to here. So when we read from here to here, what is going to happen? We feel that AUG, okay, methionine, 1. UUU, phenylalanine. So repetition of UUU is there, so we just consider it as second amino acid. And the third one, basically we think that UAA is what? The stop codon. So we think only 2 types of amino acids are being formed, which is why we go with C option. But listen to me carefully. You have to think ulta. You have to think ulta, right? So when you think ulta, what is going to happen? D. Who is saying D? Okay, listen to me. When you think ulta, what is going to happen? AAU, this is 1. UUU, this is 2. Again UU repeated, again UU repeated, ignore. GUA, this is 3. So how many types of amino acids are going to be formed over here? These are 3 types of amino acids that are going to be formed over here. Your answer for this question is going to be B. Why do you think it is D? Why do you think it is going to be D? You're most welcome, Ziratar. I hope I pronounced it right. So those of you who gave me the answer B, let me check if at all MQP children are giving me answer correctly. Yes, Sanjeevani gave me the answer correctly. So yeah. And everyone who, what about stop codon? I told you, listen to me carefully. You are reading in 5- to 3- direction. In 5-3- it will be read as AAU, not UAA. AAU and then UUU. It goes like that. You get it? Which is why your answer would be 3 types. I hope it makes sense. I'll tell again. Listen to me very carefully. This is important. This is where we can mess around things. We have to do translation in which direction? We have to do translation in 5- to 3- direction. Over here, it is 3- to 5-. Can you go and read ahead like this? Which translation happens like this? In opposite direction. No translation is going to happen like this. So you have to read it opposite side. AAU. AA. It is not UAA. It is AAU. So this is going to be the first amino acid. UUU. Second amino acid. And basically is a repetition of that. Then GUA. The third amino acid. You get it? So your answer for this question is going to be 3 types. Fine? Okay. Perfect. Kashish, hello dear. Hello. Perfect. Next question that is out there, question number 3. Match the following. Alec Jeffrey, F. Sanger, Jacob and Monod and Griffith. Over here you have options that is there. Make sure you solve it nicely. Look at those options, do it properly, think around it and then only go ahead. Sanjeevani says the answer for this question is B. Okay. What about others? Vaishnavi says, okay ma'am, thank you dear. Okay, Shalam says B. Varsha says C. Option is correct. Okay. So I'm getting B and C. Ma'am, aap bahut acha padhati ho. Thank you so much dear. Vaishnavi says C, Mansi says B. So I have got multiple options. A, B and C is what I see over here until now. No, only B and C. Maximum votes are for B, so let's go for B only. Listen to me. What is one thing that you're pretty sure of? If at all you do not know anyone's name, what is one thing that you're pretty sure of? When you get options like this and you feel that I am not confident while solving this, look for the weakest one. Griffith. We know he's the one who did the transforming principle experiment. So D goes with 2. D goes with 2. I just have one option and with that I can clarify the answer is this. You get it? But still, sometimes we do not get an easy option out there, right? We have to look for the other things. So let's go ahead and let's see. Alec Jeffrey, who is there, my dear children, he was the one who gave the DNA fingerprinting method. So F. Jeffrey has too many F's and over here fingerprinting also has. That is how I remember. F. Sanger also have an F, but please remember F. Sanger also has an S, and S is for sequencing. DNA annotated sequencing is basically done by F. Sanger. Okay, now Jacob and Monod, both of them has O in them, and basically O is for lac operon. So you can remember it like that if at all it works. And Shalom, please dear, go and have your breakfast. Okay? Okay, perfect. Let's go ahead. Question number 4. This is from the question bank now. So make sure you solve it. This is something new. So everyone out there, let's go ahead. GGU, GGC, GGA and GGG all code for glycine. Which salient feature of the genetic code does it prove? Which salient feature of the genetic code does it prove? When I say multiple codons are coding for the same amino acid. Sanjeevani says the answer for this question is B. Okay. Mansi says it is B. Okay. What about others? Shalom, ma'am, would you give a break or not? So I was not planning for a break, but if at all you want, probably after principles of inheritance we can, because these two chapters I have not put too many questions out there. So if it works, if that's okay. Okay. Mansi says 4B, Shalom also says B, Jia also says it is B. B is what? Degenerate codons. Exactly. And Shankarappa is there. Hello dear. Says B. So when I say multiple codons are coding for same amino acid, the feature is known as degenerate codons. So when I say GGU alone is coding for glycine, what feature does it show? GGU is coding for glycine, UU is coding for phenylalanine. If at all you get a question like that, what feature does it show? It shows that they are unambiguous in nature. That means each codon will be coding for only one amino acid out there. Each codon codes for only one amino acid. That is known as unambiguous nature. For those of you who get confused between unambiguous as well as degenerate, do not get confused now. Okay, so when you have multiple codons, for example over here GGU, GGC, GGA, for the same codon you call this as degenerate feature. And you can remember it in a fun way that over here this guy thinks that I only code for glycine, but GGC also thinks that I only code for glycine. These guys do not know. So this is degeneracy that glycine is doing. It's cheating that this guy is doing. All the codons are actually coding for glycine only. You can remember like that if it works. Anyways, next question. In a single stranded DNA, if the ratio of A and G is 60 to 40, then the ratio of T and C is what? If A, my dear children, A to G ratio is 60 and 40, you are being asked about the ratio of T and C. Make sure you read the question carefully and then only go ahead with answering this. Also understand that how many number of A will be there, that many number of T will be there, how many number of G will be there, that many number of C will also be there. That is the general nature out there. Now you have to tell me the answer. Mansi says the answer for this question is D. Sanjeevani says it is D. Okay, Bishnoi says it is A. Shankarappa says it is A. Varsha also says it's A. Read the question carefully. Jia also says it is A. Ariba also says it is A. Read the question carefully, very carefully. Very carefully. So are you logged with A? Can I say something now? It's talking about a single-stranded DNA. In a single-stranded DNA, why do you care about how many A and G and T and C would be there? You need to understand that if your single-stranded DNA is there and I say these many number of A is there, I should not be bothered about the number of T. It can be so many. It can be so many. You get it? It has no relation amongst each other in a single-stranded DNA. If I talk about double-stranded DNA, obviously it would have made sense because how many number of A will be there, that many number of T will be there, right? You get it? So here it does not make any sense because single-stranded DNA is being mentioned, which is why your answer for this question is going to be none of the above. Got it? Good morning, Shreya. Okay, got it? This is super important. Do not mess it up. So your answer for this question is going to be none of the above. Is this clear? If yes, just can you give me a thumbs up? Can you quickly give you a thumbs up? Ma'am, I watch your old live. Should I watch this also? It's which live, dear? Which live? So recorded video on 1 marker. If at all you have watched this, you do not need to watch it again. There's a recorded video on 1 marker. In that, I have discussed all of this. Okay, Sanjeevani, Arib, and thank you, Mansi and Shreya Kohli. Let's go ahead with the next question that was there, and that is components of deoxyguanosin is. The components of deoxyguanosin is. So MCQs is what I believe. I have took a recorded video is what I remember. But if there's a live, and if it was done for pre-boards or finals, then you should not be. Gopesh, definitely. I have forwarded the questions that I needed to ask to the team lead out there. Now she has to coordinate, but yes, definitely doing that, Gopesh. Don't worry, okay? Mansi says the answer for this question is 6B. B is what? Guanine plus deoxyribose. What about others? What do you think is the answer for this particular question? Think. Important. No, Shreya, do not try to. No, hello, dear. Okay, so Mansi says it's B, Shankara says it's B, Sanjeevani says it is C, and Jia says it's B, Nol, Aaron says it's C. Shreya, please don't leave this chapter. If at all you can, watch the masterstroke video of this. It will definitely help you out. I'm telling you. Okay, components of deoxyguanosin. First of all, we need to break it down. What do you mean by guanosin? Guanosin is basically when you have the pentose sugar and you have guanine being attached onto it. This is known as what? Guanosin. This is basically a nucleoside out there. Now, you need to understand, if at all it is basically, there are two types of sugar, ribose sugar and deoxyribose sugar. If it is ribose sugar, that means a part of the RNA, you will just call this as guanosin. On the other hand, if it is deoxyribose sugar, that is involved over here, that is a part of DNA, my dear children, you have guanine attached to it, you add the term deoxy over here also. So your answer would be guanine is there plus deoxyribose, that is the sugar, that is also going to be there over here. So please understand that your answer for this question is going to be B. If at all you have made some mistake over here, please rectify it. And why it is not option number C? Because if it was basically option number C, the name should not have been deoxyguanosine. It should have been deoxyguanosine triphosphate, or basically 3-phosphate will be there. So yes, so deoxyguanosine triphosphate, things like that, that means then there will be phosphate group. Here there is no understanding about phosphate, which is why it will be just it will be just guanine plus deoxyribose. I hope it makes sense. Yes. So for children who thinks that this question is super clear, can you just give me a quick thumbs up if it works? Can you just give me a quick thumbs up if it works? Perfect. Let's go ahead. Thank you guys. Okay. A mature processed mRNA transcribed from a gene is 636 nucleotides long. Assuming if it codes continuously without untranslated regions and stop codon for a polypeptide, how many amino acids does it decode? That is a question. Sanjeevani says the answer for this question is A. What about others? What about others? Mansi also says A, J also says A, Kenchappa is there. Hello dear, Kenchappa also says it is A. Shankar says it is A, Nol, Aaron also says it is A. A is what? 212. Let's go ahead and check it out. So when you are saying that there are 636 nucleotides altogether, and you are saying that in this particular thing, in the mRNA, there is no UTR, untranslated regions, or there are no stop codons. That means it is going to go continuous. In that way, 3 codons basically come together to make up, 3 codons comes to make up a triplet codon out there, right? And basically, 636 divided by 3, if at all I will be doing, I'll get the number of triplet codons out there. How many triplet codons are going to be there? 212. And 212, each triplet codon, my dear children, each of them are going to give rise to 1 amino acid. So can I say 212 codons are there? That means 212 amino acid will also be there? Yes. Only when there is no stop codon mentioned over here. If at all there is no mention about anything, you have to assume that there will be stop codon. Okay, and you have to minus 1 amino acid. One triplet codon is not going to code for anything, it's just going to be a stop codon. So please understand that here, since that is not been mentioned, your answer is going to be A. Now, question number 8. What do you think is the answer for this particular question? By the way, whoever is watching this video, I hope you guys have clicked on the like button of this video. Yes? Please make sure you do that. Yeah. Next thing. Question number 8. Statement number 1. Deoxyribonucleotide triphosphate acts as substrate during DNA replication. Statement number 2. Deoxyribonucleotide triphosphate also acts as a source of energy in the process of DNA replication. What do you think is the answer for this particular question? Sanjeevani says 8A, Mansi also says A, G also says A, Nol, Aaron also says it is A. Kushi also A is what? Both statement 1 and 2 are correct. Why it is correct? Listen, deoxyribonucleotide triphosphate, it looks like this. So this is the ribose sugar. Over here you have whatever nitrogenous base is there. On the 5th carbon you would be having, my dear children, 3-phosphate group being attached. Triphosphate, that is why you call this triphosphate. Now whenever you want to make a nucleotide sequence, you want to make up a DNA, you need multiple nucleotides like this. So multiple nucleotides come into the picture, my dear children. Again, nitrogenous base over here. On the 5th carbon, what do you have? You have triphosphate. Listen to me. When you want this portion, that is 3rd carbon and this guy to fuse together, okay, that is, this guy also wants to make a bond over here with the phosphate, and when it happens, my dear children, it breaks up with the other phosphate groups out there. So 2 phosphate moves out and only 1 phosphate remains over here. So what is the basic unit of making the DNA? It is this only. It is a substrate during DNA replication. Deoxynucleotide triphosphate, it is actually a substrate over here that we need. And my dear children, here it is also said that it is source of energy. Why do you say that? Listen to me. When this bond is being broken, when the phosphate bonds are being broken over here, there is huge lot of energy which is being released, and this energy allows in the formation of the other bonds as well. That is why we say that it is a source of energy as well. So can I say both the statements are correct? I will be definitely saying that, which means that your answer is going to be A, just like how you said that. Yes? Perfect. My children are doing super awesome. Okay, let's go ahead with the next question out there. In pea plants, each character is governed by a gene, which is what? Each character is governed by a gene, which is what? In pea plant, each character is governed by a gene, which is monocystron, polycystronic, operon, or intron. What do you think is the answer for this particular question? Sanjeevani says answer is A. Oh my God, Sanjeevani is in full josh today. Monocystron. Okay, what about others? Kushi says it is B. Okay. Polycystron. I've got multiple answers now. Mansi says it is A. Okay. 2 votes for this. What about others? What about others? Baswaraj says A. Gia also says A. Okay. A. Multiple votes for A only. So I will go with A. Listen to me very carefully why it is A. Monocystron means, you first of all, by any way, if at all you're watching this video, please know the difference between prokaryotic and eukaryotic transcription, translation, everything out there. Very, very important. Now listen to me. There is eukaryotes and there is prokaryote. Eukaryotes are there and prokaryotes are there, right? Eukaryotes will be having what? The cell membrane bound cell organelles and everything is going to be there. This guy does not have all this thing. This is the basic thing that we need to know. Yeah. So genetic material is just lying out there, right? Now over here in the case of eukaryotes, what is going to happen is that they have one cystron, and by one cystron I mean one single unit of gene out there, and this gene is going to code for one protein. When I say it's one cystron, that means it contains just one gene, and this one gene is coding for one particular protein. In the case of prokaryotes, my dear children, they have polycystrons, as in basically suppose this is a segment of a gene. Here, this is one cystron, this is second cystron, this is third cystron. One cystron codes for one gene, another cystron codes for another gene, third cystron codes for another gene. They have multiple cystrons, which in this condition is known as polycystronic condition. What do you call this condition? Polycystronic condition. So if at all you have seen lac operon, you might have seen ZYA, 3 of them are together. And in one particular unit, 3 genes or basically 3 proteins are going to be formed, right? That is how prokaryotes work. So P and P, always remember P and P. Now here it's pea plant, and I know that is also a P, but please understand that when you say pea plant, P is a plant and plants comes under eukaryotes. Only bacteria are the one that comes under prokaryotes. Apart from it, everything is going to be monocystronic in nature, which is why your answer would be this. Kushi, we will be taking a break after this thing. Principles of inheritance and variation, okay? Done? Deal? Otherwise, we will not be able to complete it. That's why. If it's clear, guys, give me a thumbs up, quick thumbs up, and I'll be going ahead to the next question. Yes, perfect. Thank you. Okay, now which of these are correct? UTR, untranslated regions, are located on the DNA. UTR is present both at 5' end before the start codon and also at the 3' end after the stop codon. UTR provides stability to the mRNA. UTR is required for efficient translation. What do you think is the answer for this particular question? You have the options like this. You have to choose from this. What do you think is the answer for this particular question? Very important thing. Think. Super important. 10D. Sanjeevani says the answer for this question is 10D. Okay. What about others? Kushi, yes, this is for second PUC boards itself, dear. Gia says the answer is D. Okay. What about the other children? Oh my God, everyone slept. Come on. Good morning. Kukuduku. Wake up, guys. It's exam time. We cannot go lazy over here. Come on, your teacher is super energetic in the morning. You know what? I am definitely not a person who gets up early, but today I did for you guys. So make sure you make the best use of it. Vinayak, hello dear. Okay, so your answer for this question, I get only 2 answers. Okay, Vinayak also gave me the answer. He said the answer for this question is D. D is what? D is 2, 3 and 4. 2, 3 and 4. Let's see. So UTR are located on the DNA. That will be wrong. UTR, untranslated regions, are there on the RNA, mRNA. It's not on the DNA. So this statement is anyway wrong. UTR is present at both the 5' end. So this is the mRNA, I make the 5' end, and this is the 3' end. Listen to me carefully. UTR will be present over here before the 5' end and after the stop codon, that is at the 3' end. You should know that here you have the start codons and here you have the stop codons. So when they say that UTR is present at the 5' end before the start codon, this is correct. When they say that it is present at the 3' end after the stop codon, this is also correct. Now, UTR provides stability to the mRNA. Do they? Yes, that is one important feature of UTR. Untranslated regions, they are not going to get translated, but they provide stability, and they are the ones that helps in initiation of the whole process of translation. You should know that. So this is also important. Now, UTR is required for efficient translation. Definitely yes. So your answer for this question becomes what? 2, 3 and 4 are correct. So this will be D. And my dear Sanjeevani, Jia as well as Vinayak were absolutely correct when they said that. Okay, perfect. Let's go ahead with the next question out there, and that is, in a DNA strand, if there are 100 nucleotides and 30 are adenine, the number of thymine, guanine, cytosine respectively are. If the DNA strand, there are 100 nucleotides and 30 are adenine, the number of thymine, guanine, cytosine respectively are what? Sanjeevani says the answer for this question is A. A is what? 30, 20 and 20. What about others? Mansi says A. Okay. Still others, I'm still waiting for you guys to tell me the answer. What do you think is the answer? So when I say DNA strand, I'm assuming it is double-stranded. Since they have not mentioned a single-stranded, obviously I'm assuming it is double-stranded, right? Now when I say it is having 100 nucleotides and adenine is given to be having 30 of them, thymine should also have the same then. If adenine is 30, that memory of thymine will be there, so 30. What about guanine and cytosine? What about guanine and cytosine? Listen to me. 30 plus 30 is equals to what? 60. Out of 100 of them, 60 of them are these guys only. Minus if I do, I get 40. Now 40 nucleotides are still left and I have to divide between both of them. 40, how will I divide? I'll make 20 over here. If guanine is 20, cytosine is also 20. Now you have been asked in sequence, you have to give the answer in sequence only. Sometimes we mess around there. Thymine is the first guy that is being asked. Thymine is 30. Guanine is 20 and cytosine is also 20, which is why your answer for this question becomes A. Awesome. Awesome, Sanjeevani, Mansi as well as Jia. Perfect. Okay, now let's go ahead with the next question out there. Done. We go ahead with the next chapter altogether, and that is Principles of Inheritance. So as I said, I just mixed and matched this chapter before that. Okay, so I hope you guys do not mind it, and let's solve the next chapter's question. Ready? Everyone super ready? Give me a thumbs up. I think it's unique, guys. Need a break. We'll soon do that, okay? These chapters also do not have too many questions. We'll solve it and then we'll take a 10 minutes break and then we'll come back. So, Vinayak, Sanjeevani and Mansi, here we go. Okay, this question we also should be able to solve. Progenies with AB and O blood groups are not possible from which of the following mating parents? This is from your model paper. From your model paper. What do you think is the answer? A, B, C, D. What do you think is the answer? 1A, 1A, Sanjeevani says 1A, Vinayak says A, G also says A. A is what? O into AB. So this is what will not give you AB and O blood group at the end. This is very simple. I'm telling you again. I've explained to you before, but there might be children who might be watching this live later on. So listen to me carefully. If you want AB blood group, you need from one parent IA, another parent IB. And you do not want that to happen, right? You do not need AB blood group. So you have to make sure that whichever parents have A on this side and B on the other side, they have to eliminate it. So A is here, B is here, eliminate them. A is here, B is there, eliminate them. A is here, B is here, eliminate them. The only answer that you have is O and AB. That's all. As simple as that. Next question. What is the possible blood groups of an offspring when both the parents have AB blood group? When both the parents have AB blood group, what is the possible offsprings? What are the possible offsprings that a parent can have? Sanjeevani says B. What about Vinayak? Vinayak also says B. What about others out there? Mansi says B. Vinayak says B. Okay. So since you guys are saying B, let's lock B. Over here, both the parents are having AB blood group. So obviously the combination is something like this. When this and this comes together, I get IA. When this and this comes together, I get IB. That means AB blood group. Here I got A blood group. And when this and this, again IB. That means again AB blood group. And finally when this and this, I get AIB and IB, which is B blood group. So B blood group, AB blood group and A blood group is what I get. So your answer for this question is going to be this. Okay. It's Habiba. Dear, I have taken questions from the model question paper and also from the question bank. So first few questions will be from, what do you call, in every chapter, first two questions that you see would be from MQP, as you can see, and rest everything would be from the question bank. Bushra, yes, please ask me loud. Genes located very close to one another on the same chromosome tend to be transmitted together and are called as what? Genes which are very close to each other, okay, and basically wherever they go, they are going to go together itself. What do you call these type of genes as? So whenever suppose you have one more chromosome over here and there is crossing over, this part goes this side, this part goes this side, whatever you do, my dear children, the genes will be remaining close only. What do you call these genes as? Tightly linked genes. So your answer would be, Sanjeevani says 3C and so does Mansi. Linked genes is your answer, and not just linked, they are tightly linked over here. Okay. Tightly linked genes shows lesser recombination. Loosely linked genes shows higher recombination. Okay. Yes. Yes. Perfect. Try it out. Everyone try it out. Which three scientists independently discovered Mendel's work? Try it out. And by then I'll solve a question. The whole year I have prepared for NEET. Now I am afraid of boards. Bushra, listen, when you have prepared for NEET, that means you are very thorough with your NCERT, or at least you might have tried your best to have an understanding of your NCERT. In that case, it should not be very tough for you. Go ahead with the questions. Are you someone who is from Karnataka? Are you going to give second PUC examination, or are you from CBSE boards? For CBSE, I think board examination has already been done, right? Now, if you are from second PUC boards, there are multiple videos that I myself have made. You can just watch those videos. There is one video which is there where all the questions are there. Like, I do not, second PUC, yes, Bushra. So there is a video in the channel, if at all you search for it, look for it, where all the 1 marker, 2 marker, 3 marker and 5 markers are being merged together and put. It comes out to be 10 hours. But I am sure if at all you give those 10 hours for that video, each and every concept that is super important will be super clear. You do not need to know, I mean, you can just be safer on that particular side, and you can just trust me on that. Okay, I'm telling you, you can trust me on that. So if you want to go the traditional way of basically dealing with one chapter, one by one, and reading the NCERT and then doing the question, you can do that way also. Because in that video, it is given chapter wise. But please do that. It is definitely going to help you out. Yeah? Arib, dear, I will be giving it to you, okay? So it was there in the previous drive link, so it was given before. So yes, you can just go through it, and Bushra, and yeah. So Bushra, I can give you the link for that particular video, mostly towards a break or something like that. Yeah. Okay. So Arib, I will be doing that, yeah. And my dear children gave me the answer for this question. D is what you answered. D is what? D, Rice, Correns and Shamak. Exactly. These guys independently rediscovered Mendel's work. And my next question would be, which year did they do that? Any idea in which year did they rediscover Mendel's work? In which year did they rediscover Mendel's work? A question to you guys. 1900. Perfect. One of yours there. It is 1900 where they rediscovered Mendel's work, and then 1902, two guys came in to give the chromosomal theory of inheritance. Okay. A plant having the genotype A, capital A, small a, capital B, small b, will produce dash kind of gametes. What do you think is the answer for this? What do you think is the answer for this particular question? a plant having the genotype this will produce dash type of gametes. Mansi says 4B is the answer. Okay. What about others? Mohan says fourth. Okay, fourth as in this is what I believe. What about others? Acha, 4 is what you're saying. Okay, fine, got it. Shalam says 4B and Jaya says 4B. Sanjeevani, where are you, dear? Okay. Most of you are saying 4B, which is why we shall look into it. So whenever you have given a genotype like this, understand, there is a formula, and that is 2 raised to the power n, where n stands for number of heterozygotes. What does it stand for? Number of heterozygote, right? So how many heterozygotes are there? Capital A, small a. These are heterozygotes. Why? Because no two alleles are not the similar ones. If it was capital A, it would have been similar. But here it is not. So this is one heterozygote. This one is the next one. So 2 of them are there. So if I replace n with 2, I get the answer 4. And these many number of gametes can be produced, so your answer is going to be B. Your answer is going to be B. Yes. Sanjini, you have already given the answer. Oh yeah, sorry. Sorry, yes. Okay, so next thing, 5th question. Which of the following is not a pair of opposing characters Mendel studied? Which is not a character that Mendel studied? It's an easy way to put that question up. Okay, which is not the pair of character that Mendel had studied? Axial and terminal flowers, full and constricted pods, green and yellow pods, pink and white flowers. D. Jia says, oh, so Sanjeevani, okay, wait, I have to go up. Mansi says D, Sanjeevani says D, Jia says D, Mohan also says D. Ma'am, you teach physics also. To be very honest, I'm really very bad at physics. I have not touched physics from so long now, but yes. Yes, so D. Arib also says the answer for this question is D. Mohan, Jia, everyone says D. So D is what? Pink and white flowers, because there were no pink flowers, my dear children. There were violet and white flowers, okay? And violet was the dominant one and white was the recessive one. So violet was the one which is the thing and pink is not there, so which is why this is not in the thing. Now, going ahead, chromosome maps or genetic maps were first prepared by? Who prepared the genetic maps? Who prepared the genetic maps? Sutton and Bovary, Bateson and Punnett, Morgan or Sturtevant? What do you think is the answer for this question? Okay, Mohan is saying D, Mansi is saying D, and Sanjeevani also says D is the answer. Sturtevant is what you're saying? Sturtevant. So listen to me carefully. What do you mean by genetic mapping? Genetic mapping means, for example, this is a chromosome and I'm saying that this particular gene is over here, or probably this particular gene is over here. I am mapping the chromosomes and basically I'm saying the genes are located at this particular position. This is known as genetic mapping. Who gave an idea of it? You know, there was a guy whose name was Thomas Hunt Morgan. He was the one who came up with linkage and recombination. He said that basically if two genes are far away from each other, suppose it is capital A, capital B, and this is small a, small b, if they are far away from each other, they will show more recombination. How? So basically, if suppose this part goes to this part goes to this, I get a new combination. Capital B, sorry. Capital B is still over here, small b is this over here. You have to interchange this. You have to interchange it over here. So you see, new combination is being formed. Here also new combination is being formed. Why is it happening? Because they are far away from each other. So whenever genes are located far, they do more recombination. So if I know the percentage of recombination, that is suppose I am saying 37.3, that means it is too much recombination. 50% is the highest amount of recombination out there. And when I say it is close to 37, it means it is too much. With the help of the data from Thomas Hunt Morgan's things, his student Alfred Sturtevant came into the idea that, okay, if I know the recombination data, probably I know the percentage of it, that means they are probably this far away. If they are this far away, probably they might be located at this particular location. That is exactly what they did. You get it? So it was a teacher-student relation that worked out really well in this particular case, and Sturtevant was the guy who came up with genetic mapping. Okay, linkage and recombination was given by the teacher, who is Thomas Hunt Morgan. So my dear children, can I also believe that one day you guys will also make me super proud? But well, to be honest, you guys make me super proud every single day. So you not have to do something super extra, but give your best for the next examination, whatever it is, right? So that is the only thing that I can ask, and yeah. Oh, okay. Next, Which of the following is incorrect regarding ZWZZ type of sex determination? And your options are this. Sanjeevani says C, G also says C, Mansi says C, Sanjeevani says C again. Okay, Anand is also here. Hello Anand. Says C is the answer. B. Okay, B. Yeah. I'll score out of out. Yes, Swaraj. Wishing you the best. Arif also says C. C is what? C is the females are heterogametic and males are homogametic. Let me see. You have been asked what is incorrect. Always remember when you get a question like incorrect, correct, underline the term there. Which is why because you might mess around things out there. Okay. So listen to me carefully. Incorrect is being asked and ZWZZ is being asked. Listen, this is the only case where the females are going to be heterogametic. Females are going to be heterogametic. They have two types of gametes, Z and W. Males, on the other hand, will produce same type of gametes over here. Got it? And you can remember this by W for women. So this is the only case where females are heterogametic. So your answer, if you're saying it is C, females are heterogametic and males are homogametic. Do you think that is incorrect? That is correct. No, you have been asked about what is incorrect. Females are homogametic and males are heterogametic. This is going to be incorrect. Your answer is going to be B. Okay, your answer is going to be B. I saw. No, it is B. Okay, so Mohan and Sanjeevani, I think these two people were the ones who gave me the correct answer. Rest everyone, I think we all did the same mistake initially. We look for the correct statement, right? Hi ma'am, I'm Shifa. Hello Shifa. Yes, perfect. So yes, this is how we become more careful towards the just a second. Okay, next one, question number 8. A woman with normal okay, a woman with normal vision whose brother is colorblind marries a man with normal vision. Oh my God. Okay, listen. A woman is there with a normal vision and whose brother is there, he's colorblind, marries a man. So woman marries a man with normal vision. So both women as well as the man is having normal vision out there. Okay, they have 4 children, 2 sons and 2 daughters. One son is colorblind while the other son and both daughters have normal vision. The pattern of sexual inheritance of the disease is, what do you think is the answer? Now, the first part of the question is just to confuse you guys. The question itself gives you the answer. Why? Why do I say so? Because over here clearly they have mentioned color blindness, and if at all you know it. Color blindness is what? Sex-linked recessive disorder. Only 2 sex-linked recessive disorders are there in your book. One is color blindness and the one is hemophilia, right? Yes. And everything in your book is recessive. Sex-linked, all recessive. Okay, 2 of them are recessive. Autosomal, everything is recessive. Only one is dominant, and that dominant one is not explained also. It is given in the form of pedigree chart, and that is myotonic dystrophy. That is the only autosomal dominant disorder. Apart from that, every other thing is recessive. Always remember that. Your answer for this question is going to be A. Next question. If both parents are unaffected carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? If both of the parents are unaffected carriers of thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? Come on, guys. Ma'am, talk with us. Yes, dear, I am talking to you guys. Sanjeevani says it is A, Mansi says it is A, Mohan says it is A, Gia also says it is A, and Mansi 9th question now. Is this 9th question? Yes, we are solving 9th question. Guys, are we solving 9th question or 8th question? 9th question, Gia says, so those of you who have given me 9, I am reading out again. Mansi, Gia, and Sanjeevani has a different answer this time. Mohan has a different answer, A. Okay, listen to me carefully. Let me try to solve it. So, it is autosomal, you know that. Yes, and now we have to think. So, whenever T means thalassemia allele is there. Okay, why I have made one HB normal and one HBT? Because here it is written they are carriers, and when they are carriers, that means they have one gene which is mutated, other one is normal. Okay, and since it is a recessive disease, it will only appear when both the genes will be affected. That's when it will be affected. Otherwise, they will be unaffected. So this is how it goes. Now let's try to cross. This and this comes, I get HB, HB. This is very normal. This is normal person. This and this comes, I get HB and HBT. This is going to be again what? A carrier. This and this comes, I get HB, HBT. Again, this is going to be a carrier. Why? Because the normal allele is there. When this and this comes together, I get HBT and HBT. So out of 4 children, 1/4. 1/4 means basically what? 25%. Your answer is going to be A. Did you get it? So everyone who said answer is A, you guys are correct. And dear Sanjeevani, I hope child you have got it. And Baswaraj, man, can we solve 3 papers plus preparatory? How much marks can I expect? I cannot tell that, dear, because in biology the trends have always been different. I'll just request you to solve the model question paper. Do not leave it. Solve the model question paper, but along with it, please practice newer questions from the question bank. Super important. Okay, please do that. Yes, perfect. For mathematics, dear, there will be a live session conducted by Ankit sir in the afternoon. Okay, so next one we have human health and diseases. And how many of you need a break? Shall we take a 10 minutes break and come? 10 minutes break. Me. Okay, so let's take a 10 minutes break then. So I do not want to torture you any further. Kit Kat break. Meet at 10:55. Be right back. And when you come back with a lot of your friends, okay? Bye-bye. Okay, champions, I believe that I am audible and it's time to go ahead with the next chapter out there. Hello, Sanjeevani. Whoa, what punctuality. So we'll just wait for some time so that others also can join, and then we'll go ahead. Hello, all type gamer girl. Hello, 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 dear. Okay, I think your time is precious, and which is exactly why it's Ritika ma'am. Thank you so much for giving me that short name. Hello, Mansi. Hello, Jia. Okay, let's go ahead with the next chapter, that is human health and diseases. And Ammu, hello, dear. Okay, let's go ahead with the first question that is out there. This is from MQP, obviously. Then we'll come ahead with question bank questions as well. So, human immunodeficiency virus replicates in what? We have solved this question before. B cells, NK cells, helper T cells, and PMNL. What do you think is the answer? Sanjeevani says the answer for this question is 1C. What about Mansi? Mansi also says it is C. C is what? Helper T cells. What about others? Any other answers? It's Habiba. Says it is C. Okay. Prajwal says C. Jia Danawar also says it is C. Okay, C is what? Helper T cells, and this is exactly the answer. Listen to me always carefully. HIV virus which is there, its actual target, okay, the target which is there, it is helper T cells. Why? Because this guy is the one that helps and increases our immunity. So if this guy is sorted, cleared out, obviously our immunity is going to go low, and that is exactly what this guy wants, HIV wants. Now, listen to me carefully. Over here, when it enters our body, it actually enters the macrophages first, and it replicates out there, and that is why we call the macrophages as what? HIV factory. But here macrophage is not an option, which is why we'll go with the next option that is there, helper T cells. I hope that's clear, but please know which is known as the HIV factory. HIV factory is this. Yeah? Perfect. Now go ahead with the next question. The drug which is a depressant and slows down body function is what? The drug which is a depressant and slows down body function is what? It's Zaiba says also T helper cells, and Mansi, Prajwal, Diya, MD, Yasin, hello dear. Okay, why not you is there, Udupal is there. Okay, so you guys are saying the answer for this question is A. A is what? A is SMAC, depressant and slows down the body function. What is the actual name of this particular drug? The scientific name of this particular drug? I told you previously, it is known as what? Heroin. It is known as heroin. And the other name for heroin is what? SMAC. And SMAC is actually a depressant and it slows down your body function. Exactly. So it is not heroin, okay? You have to remove the E out there. It is just heroin. And can you tell me the other name of heroin? The other name of heroin, it is there, but the scientific name of heroin is different. Can you tell me that? It's okay, guys. Don't retract. It's okay. I was just making fun. Diacetylmorphin. Exactly. It is also known as diacetylmorphin. Why do we call this guy like that? Because initial product is morphine that we get from papaver, and then we get heroin. How? When it undergoes acetylation, that is how we get heroin out there. So your answer for this question is SMAC or diacetylmorphin. I hope you have got the answer. Samiha, hello dear. And Sanjeevani, you were the person to give me the answer, and also Samiha, good, good, good. Venkatesh, SMAC, yes, SMAC. Now the next thing, this is how you get it. Well, this is papaver somniferum. The latex of poppy is from where you get the morphine. Morphine undergoes acetylation, and that's how you get that guy also. Okay? Yes. Now, next question. Symptoms of constipation, abdominal pain and cramps associated with the disease what? What are the, these symptoms associated with which of the disease out there? Ma'am, are you solving now only MCQs? Yes, dear. I'm solving only MCQs over here. And yeah. 3. Amoebiasis. Say Sanjeevani, and Itzaiba says also amoebiasis. Mansi also says amoebiasis. Amoebiasis, do you think that is the answer for this question? Symptoms of constipation, abdominal pain and cramps? Yes. All of this together when it comes, it is amoebiasis. It is also known as amoebic dysentery. And yes, Udupal, yes, MD Yasin, Ammu and Venkatesh, everyone. Awesome. Okay. So that is actually correct, and it is also known as amoebic dysentery. Perfect. Now the next question that we have is anti-venom given against snake poison contains. So suppose someone is bit by a snake and they go to the doctor and the doctor says that, hey, Macha, you wait, Macha, your body will release antibodies and you will be safe. Does the doctor do like that? No, Baba, the doctor gives anti-venom. What does this anti-venom contain? Yes, fourth C is what? Antibodies. Say Mansi says, ABHYAS says antibodies, Sanjeevni, MD Yasin, and Ammu, Udapal, Samia, Venkatesh. Why not you? Oh my God, awesome. Answer is antibodies. It's just directly antibodies. You call them preformed antibodies. What type of immunization is this? This is passive immunization. Why? Because your body is not actively producing antibodies. Rather, you're depending on a passive source out there. Right? Yes. Now next thing, the substance produced by a cell in viral infection that can protect other cells from further infection is substance produced by a cell which is undergoing viral infection, my dear children. So there is a cell which is there, Papa cell, okay. This cell is Papa, why Baba? Because it is infected by virus. But this cell release certain things and tells the other cells, Macha, you guys be safe. Macha, you guys be safe. What does it release over here to instruct the other cells to be safe? It is interferon. Yes, exactly. You guys are correct. C answer is what? Interferon. This is coming under which type of barrier? Come on. This comes under which type of barrier? When I say interferon is released over here, it comes under which type of barrier out there? Physical, physiological, cellular, or anything else? Cellular barrier, nope, 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 nope. Physical, think, Macha, think. Listen, always we have to should not be mugging up things. We need to think about it. Physical as in your skin and everything, that is a barrier. Physiological means secretions of your body, that is saliva, tears and stuff like that. Cellular means cells which are involved directly to kill the other cells. For example, you have neutrophils, or that is known as PMNL, or probably natural killer cells. Now there is one more, and that is cytokine. Exactly. So okay, who gave me the answer? Why not you gave me the answer? Cytokine, and also Sanjeevni gave me the answer. YD Yasin gave me the answer. Samiha and Jigar gave me the answer. For those who did not give me the answer, that means that we just need to study that particular concept. That is our important concept. So what do we need to do? In a piece of paper, write it. That barrier method, I just have to go through it. Okay, that is how we basically rectify things, right? So it's a cytokine barriers. Whenever cells are releasing certain things and that acts like the protection, so the cell is releasing interferon, right? So that is the reason your answer would be this. Ma'am, can you please upload answer PDF? Arib, I will be doing that, definitely, for sure. Promise. Okay? Yes. Next thing, dash is a very effective sedative and a painkiller. Dash is a very effective sedative and a painkiller. Sedative means you feel sleepy. Okay, sedative means you feel sleepy. So what is the most effective sedative and a painkiller? Any idea? Just a second, okay? Morphine, ma'am. Okay, morphine, ma'am, morphine. Okay, morphine is the answer. Morphine is definitely a sedative and a painkiller. And from there you get the heroin out there. Now, next question, this is from the question bank. This is not from your MQP, so you have to think. Okay, you have to think for sure. So, yes. Which of the following pairs correctly matches a disease and a pathogen causing it? Typhoid, salmonella typhi, pneumonia, Haemophilus pneumoniae, malaria, Ascaris lumbricoides, ringworm, and amoeba histolytica. What do you think is the answer for this question? 7A, Jigar. Yova is there. Hello, Yova. Lakshmi says, ma'am's PDF. I will be giving you PDF post this lecture, okay? Why not you says A? It's Saiba also says A. Sanjeevni says A. And then whom I have got? Udapal says A. Samiha also says A. Shivalinga have retrieved the message, okay. Yasmin also says A. Okay, perfect. Ringworm. Ritika, okay, listen to me carefully. Which is correct match, no? Typhoid is caused by salmonella typhi only. Typhoid, name things only. It's very easy. So this guy is the correct answer. Pneumonia, Haemophilus pneumoniae. This is mostly where we do mistakes. Listen, Haemophilus influenzae causes pneumonia or Streptococcus pneumonia causes pneumonia. But here it is ulta. Haemophilus is taken from here, pneumonia from Streptococcus pneumoniae. So it's a mix of both. So this is why it is wrong. Malaria is caused by Plasmodium. It is protozoa, not by Ascaris. Ascaris lumbricoides cause Ascariasis. And finally, ringworm is is caused by, ringworm is basically what? It's a fungal disease, but it looks like a worm shape on our body. That is why we call this ringworm. But it is not caused by worm, it's caused by fungus, and Entamoeba histolytica is the one that causes amoebic dysentery. So these guys do not pair up. So your only answer for this question is A. By the way, everyone out there, give me this particular thing. Who or which fungus causes ringworm disease? I have taught you in the MQP, I have told you a way to remember it also. Please tell me the names of the organism that causes ringworm. I'm telling you, you are going to get a question based on the infectious diseases and all the symptoms and things like that. So you have to be pro at that particular topic. Okay, you have to be literally a pro at that particular topic, so make sure you do that. Trichophyton rubrum. Yes, this size MGD, you got it correct. Trichophyton epiphytona. Why not you trichophyton? Okay. Spelling, check. Okay. Spelling, guys, you have to check your spellings. Microsporum trichophyton epidermophyton. Jigger, perfect. Samiha, trichophyton, exactly. A way to remember this is basically, listen, it's our skin or basically the scalp, everything which is affected. So always understand these two guys, trichophyton and epidermophyton, they are fighting. Fighting for what? A small spore that is known as microsporum. So microsporum, trichophyton, epidermophyton, those are the causative organisms for ringworm disease. You can remember like that. Okay, if at all you want. That would definitely help you guys. Yes, yes. The next thing that we have is hemozoin is what? Hemozoin is what? By the way, everyone who gave me the answers somehow the other way, perfect. You are doing absolutely great. Now, hemozoin is a precursor of hemoglobin, toxin released from streptococcus infected cells, toxin released from plasmodium infected cells, toxin released from hemophilus infected cells. What do you think is the answer for this particular question? 8C. Mansi says 8C, Mohan says 8C, Sanjeevani says, Yova also says C, Jigger says C, and MD Yasin, Udupal, and all-time gay, okay, Ritika, okay, Samiha, Dhruti Gowda is also here. Everyone says C. C is what? Toxin released from plasmodium infected cell. I hope you guys know that. Hemozoin is basically nothing but our RBC. In our RBC, what do you have? You have hemoglobin. And suppose now this pathogen is there. This particular pathogen is there whose name is what? Plasmodium. It actually wants the globin. From hemoglobin, it actually wants the globin. So what does it do? It breaks it down. Heme to heme as well as globin. But when it sees heme, it got all scared. Why? Because, my dear children, heme is very toxic for the plasmodium. So what does plasmodium do? It converts heme into something else which is known as hemozoin. Hemozoin. Now, hemozoin is not toxic for this guy. But when RBCs are finally going to divide open, okay, these hemozoins are going to be released into our blood and we are toxic to this. Okay, or not we are toxic to this. Hemozoin is toxic for us, which is exactly why we get fevers and chills. Okay, so it is toxin released from plasmodium infected cells. I hope this is super clear to you. It converts to hemozoin. Exactly. Yasmin, everyone's doing super great. Let's go ahead. Here is the basic thing about plasmodium. I hope you guys know about it. I'll ask you certain questions. Can you give me the answer? If at all you give me the answer, I'll think that, okay, you have understood, you know things about malaria. In which stage does malarial parasite affects us? In which stage does malarial parasite affects us? What do you think is the answer for this question? In which stage malarial parasite affects us? Sporozoid. Jigger says sporozoid. What about others? What about others? Is it sporozoid stage? Yova also says sporozoid stage. So listen to me. It is sporozoid stage. Samiha also says sporozoid. Sanjeevani also says sporozoid. Okay, so suppose I am the mosquito and in my salivary glands I have the sporozoid stage. When I go and drink the blood, basically I release my saliva. Why? Because the saliva has anti-clotting enzymes such that I can drink the blood very easily. So with the saliva goes the sporozoid inside the body. Okay, now in which stage, I'm asking you the question, listen to me out clearly. In which stage does the mosquito take back the blood? So when the mosquito comes to drink our blood, what stage is transferred from us to the mosquito? What stage is transferred from us to the mosquito? So it entered the sporozoid stage, but obviously it will change up in our body. Yes, Jigger says gametocyte. Hi, Syed. Jigger says gametocyte. What about others? Any idea? In which stage does the mosquito take up the pathogen out there? Yova also says gametocytes. Yes, it is the gametocyte stage. So it enters in sporozoite stage and later it undergoes division, and finally, my dear children, it is going to form gametocyte stage, and these gametocytes are being there in our blood, and from our blood the mosquito will be drinking it up. Okay, Sanjeevani also says gametocyte. Good. Ma'am, 5-mark questions coming application level, so that easy method to answer. Shivalinga, I didn't get your question exactly. So the 5-mark questions we have dealt separately, and my dear child, if at all you have seen that, that is definitely going to help you out. Okay, read the question. For understanding all the application-based 5-mark question, you need to know the concepts really strong. Make sure you do that and practice from question bank. In the question bank, there are some nice level of questions. Okay, I have included in the 5-mark video. If at all you want to, you can watch it. Okay, which of the following is an autoimmune disease? Rheumatoid arthritis, tuberculosis, malaria, or AIDS? 9A, Jigar says 9A, Mansi says A, Samiha says A, Sanjeevani says A. Okay, Ritika also says A. A is the answer. Rheumatoid arthritis is what is an autoimmune disease. Autoimmune disease means your body does not understand which is our cell, which is not, which is why it goes on killing all the cells out there. Very scary, right? So, yes, A. Next one, the site where lymphocytes interact with antigens and proliferates to become effector cells are. The sites where the lymphocytes interact with the antigens and proliferate to become effector cells. What do you mean by that? If at all you know about the journey of lymphocytes, my dear children, there is one very important thing that you need to know. There are primary lymphoid organs and there is secondary lymphoid organs. In the primary lymphoid organs, these the B and the T lymphocytes are going to be formed. Okay, they are going to be formed. They mature over here, and not just that, they become antigen sensitive. They become antigen sensitive, it means that they know that I have to basically kill this antigen in future. In future, do they just start killing over here itself? No. And what are these primary lymphoid organs? It's bone marrow and also thymus. These are the primary lymphoid organs where they just become antigen sensitive. Where do they become effector cells? Effector cells means finally they are going to show their action. They're going to kill those antigens out there. So those are secondary lymphoid organs such as lymph nodes, spleen, Peyer's patch, tonsils, MALT, all of it. Okay, so your answer for this question would be, so you are being asked about where do they become effector cells? That means you have to look for the secondary lymphoid organs. Thymus is a primary lymphoid organ, cancel out. Bone marrow, primary lymphoid organ. Bone marrow, primary lymphoid organ. You just have this option, that is Peyer's patch and tonsils. C is your answer for this. So who gave me C answer? Let me see. I knew you will get it wrong. Okay. Samiha and Sanjeevani gave me the answer, and also Jigar has given me the answer. My dear children, make sure you check for this. This is very important concept. We make mistake. The terms are super important. Here they become antigen sensitive. There they become effector. Okay, here they become antigen sensitive. That means they just know the antigens. They don't work against it. Here is the place where they actually do that. Okay, perfect. Let's do the next question. Injection of antitoxin in tetanus confers which type of immunization? So, okay. Nagabhushan, it's okay dear, you are, if at all you feel like that, please go ahead with someone else's video. I do not mind that. Yes, Darshan, please ask me the question, dear. Okay, injection of antitoxin in tetanus confers which type of immunization, champ? 11B, 11B. Jigar says B, Mansi says B. What about others? Richie says B, Sanjeevani says B. Okay, Richard Anthony is also over here. So most of you guys are saying B, and B means what? Passive immunization. Do we get passive immunization out there? Listen, whenever you might have seen, whenever we we go around somewhere and we fall down, okay, at that particular point of time we are asked to take but this particular thing. Okay, that is a tetanus injection. Why? Because there are chances that the bacteria can get inside your body, and against it you immediately need an antitoxin. When you get an antitoxin, basically you're getting again the preformed antibodies against this organism out there. And when you're getting preformed antibodies, that means you're talking about passive immunization. So your answer for this question is going to be B. Okay, so most of you gave me the correct answer. Actually, everyone gave me. And you guys are doing super crazy good. Awesome. Now, 12th question, let's try to solve this one. Tobacco consumption is known to stimulate secretion of adrenaline and noradrenaline. The component causing this could be what? Ritika, but I like your voice. Thank you, champ. And Darshan, model paper is enough for board. Darshan, I will not say in bio you will get direct questions. You will not. One or two questions might be there, but not all of them will be there. Okay, so please make sure that you go ahead and understand the concepts behind all the questions which are there in the model question paper, so that you can solve more of them. You get it? And also solve some questions from the question bank. Yeah. Okay, so I get Jigar, Sanjeevani, MDSC, Darshan, and Richard, and Kavita, and Samiha, J and K Madi. Hello. Okay, so many children are there. Okay, everyone is saying Ritika, everyone is saying A. So A is what? Nicotine. Tobacco consumption gives us nicotine? Yes. And nicotine, my dear children, please remember, this is important, okay? Nicotine, whenever the term comes, which one is the thing or which drug is the thing that messes up with your blood pressure? Just understand it is nicotine, okay? Do not go ahead with cannabinoids. I know cannabinoids also have an effect on your cardiovascular system, but this is a unique feature about nicotine. It increases your blood pressure and things like that because it helps or basically stimulates the release of adrenaline and noradrenaline. That's the reason. So your answer for this question is going to be A. Okay? A. Perfect. Everyone. Nagabhushan. Everyone. Perfect, Macha. Okay, let's go ahead with the next question. By the way, can I ask you a question right over here? I want to ask you a question. Which is the drug that is abused by sports people? Which is the drug that is abused by sports people? Please make sure you're good at it. You just check it out. Okay, Yova, can you speak Tamil a little bit more? I am not really pro at Tamil. I'm actually a Malayali, but yes, I understand Tamil because it's kind of very much similar. Tamil konjam theriyum. Okay, I like your voice. Your teaching makes every doubt crystal clear. Thank you so much, Mansi. Because of you, I'm good at biology. I'm following you from first year. You're my favorite teacher. Oh, that means a lot. Thank you so much. So yes, cocaine, crack, cannabinoid, everyone is giving me different answers. Sanjeevani gave me the answer, that is cannabinoid. Okay. Natil, I am in Wayanad. Njan Wayanadil nanu. Okay, guys, it's the answer for this question is cannabinoid. The drug which is abused by sports people is cannabinoid. Do not get it wrong. I have taught you before in the 2 marker, 3 marker, 5 marker, any of the videos, it will be there. Definitely make sure you know these small details. It will help you. Okay, next. So I think only one person gave me the answer. Others have tried, which is why kudos to you also. Okay, now charas and ganja are the drugs which affect the respiratory system, cardiovascular system, digestive system, nervous system. What do you think is the answer for this? Cocaine is wrong. Yes, dear, cocaine would be wrong. It's cannabinoid which is abused by the sports people. It's given in your NCERT one line in the cocaine wala, the paragraph, no? If you read the, the last point over there, you will be able to see that. Towards the end, it will be there. Okay, Sahana, good morning. Good morning. And Mansi says it is B, and Kavita says cardio. Okay, cardio, that is B. Okay. JNKWD says B, Sanjeevani says B, and Sahana also says B. Cardiovascular system. Exactly. Why cardiovascular system? Because charas and ganja, both of them, they come under cannabinoids. My God, you children are awesome. Perfect. Super awesome. Next, 14th question. A chemical structure is given below. Identify its plant source. Can you tell me what is this first? And then we can go ahead with identifying this. Sahana says B. I think it was for the previous question, right? Guys, if at all you can give me the answer like this, 14 A, B, C, D, that will be easy because I will not know which question are you answering, okay? Sanjeevani says the answer for this question is C. C is what? Papaver somniferum. Aha, okay. And Jigar is asking, is it morphin? Kavita was also saying it's morphin. Yes, it is morphin. Okay, it is, this structure is of morphin, and always remember, it looks like an O, right? It looks like an O. Morphin, they come under opioids. Morphin, which is there, they come under opioids. opioids, and that also has an O over here. And where do you get the opioids from? From the Papaver somniferum. Similarly, one more diagram is given in your NCERT, and there you will be seeing something like this at the end. That is of cannabinoid. Some structure might be there, and something like this at the end will be there. That is of cannabinoid. Only two structural things are given. One is for morphine, other is for cannabinoid. So yes, Papaver somniferum, syncarpus 2, exactly. Awesome. So Mansi, JNV, and Richie, Jigar, Jia, and Yova, every one of you are absolutely correct. Before that, Sanjeevani, Richard Anthony, awesome, awesome, awesome. Now, 15th question, the disease chikungunya is transmitted by? We can recognize by port shape. Is it? Okay, that works. The disease chikungunya is transmitted by, my dear children? Any idea? Disease chikungunya is transmitted by, okay. 15th, just a second. Kavita says by Aedes mosquito, okay. Sanjini says 15B. Mansi says 15B. Richard B. JNKV B. Jigar also B. B is what? Aedes mosquito, exactly. Chikungunya as well as, okay. Chikungunya as well as this thing. That do you call, what do you call this as? I forgot. Dengue, yeah. Chikungunya and dengue, all of them are transferred through Aedes mosquito, different varieties of them, but yes. Yes, MD Vaseem, Samiha also, you guys are super awesome, correct dear. 16th question, identify the enzyme which deviates from the central dogma of genetics during HIV infection. Ma'am, slow Madi, okay, definitely, I'll slow down. Okay, identify the enzyme which deviates from the central dogma of genetics during HIV infection. What is the central dogma of genetics? Do you know that? Francis Crick gave that, he said that from DNA, you get the RNA. From RNA, you get the proteins. Okay, so from the DNA, RNA, from the RNA, you get the proteins. That is a central dogma of out there, right? Now, you have to go and think what deviates from it. C, its baby girl is also there. So I am seeing so many answers. Jigar from Jigar, JNK, Sanjeevani, Kavita, Mansi's baby girl Sahana, Richard Anthony, and Ankit Thakur. So listen, the answer for this question is C only, reverse transcriptase. Why? Because until now we saw that DNA has to form the RNA. RNA has to form the proteins. But over here, the virus which is there, it has RNA as genetic material, it enters our body, and basically from the RNA it converts into DNA. That is reverse of transcription. That is what? Reverse of transcription. For that you need the enzyme that is reverse transcriptase. So your answer for this question is going to be C. Okay, so those of you who gave me the correct answer, kudos to you. And the central dogma, one more thing is there, DNA forms DNA, that is known as replication. So replication, transcription, and translation. Yes. Perfect. Now we go ahead with the next chapter, that is evolution. Everyone understood all the questions which were there from human health and diseases? If yes, click on this particular button and send it right now over here. That is what I mean. Okay. Give me, you know what? I'm starting to forget words now. I do not know why. It's because I'm aging. Okay. Thank you guys. Okay, perfect. So now we go with evolution. I think this chapter, I have put so many questions. Yes, so let's go. Let's see. Select the correct match. Australian marsupials are on this side, and Maba, placental mammals are on this side. Placental mammals are on this side. So from this category, one guys come. From this category, one guys come. You have to tell which one is which. Mansi says 1D, okay. Sanjeevani says 1D, Kavita says D, Udupal also says it is D. So Udupal also says it is D. D is what? Spotted cuscus, and over here I have lemur. Is that so? Why is this, ma'am? I don't understand what is the question. This is usually something where children get confused. So I am here to give you an explanation for that also. Listen to me very carefully. So there is an area, suppose, okay, a geographical island where no one is there. Now, over here, Australian marsupials came into the picture. They were there over here. Some of them went that side, some went this side, some went this side, some went this side. Okay, what are they showing? Adaptive radiation. They are radiating and they are getting adapted to those particular places. Ma'am, is this an example of divergent evolution also? Yes. Why? From common ancestor, you are diverging to different areas, right? So this is an example of divergent evolution. Agreed. Now, at the same location, if at all I am saying, one more guy is also coming into the picture. That is placental mammals. From them also, few went there, few went here, and few went here. What is this? Again, they are also showing adaptive radiation and they are also showing divergent evolution. But when you talk about both of them together, these two guys who were literally unrelated are coming over here, joining at a single point. They will be having some commonness between them because they are habitating in the same area. Can I say it's an example of convergent evolution now? They were having different ancestry, they are converging to a point, showing similar character. This is convergent evolution. So you are being asked that from Australian marsupial side, if spotted cuscus came from placental mammals, who was the person who came along? That is the question, or who is the organism, not the person. So how do you remember it? There's an easy way. I've told you in the MQP session also, but I'm pretty sure some have missed it. Placental mammals on this side, Australian marsupials on this side. Okay, and listen to me very carefully. The names over here will be small, small. Here the names are going to be big, big. So if at all you spot a bigger name, definitely understand it is Australian marsupials. So mole from here, marsupial, mole from there, easy to remember, right? Anteater from here, numbard from there. Little difficult, so that is why you have to pay your attention, right? Now next thing, you have mouse and marsupial mouse here. You have lemur and spotted cuscus. The bigger name comes on this particular side. This is also difficult because it is not related. So anteater and numbard and lemur and spotted cuscus is something different. Now we have flying squirrel, flying phalanger. Bigger name comes on this particular side. Now what do you have? Bobcat, Tasmanian tiger cat. Bigger name come here. Okay, wolf, Tasmanian wolf. Bigger name comes over here. So two things you have to remember, anteater and lemur. This is something that is very unique out there. Hello, hello Gotu, and hello, it's baby girl. And hello Krishna. Okay, so yes, going ahead with the next question out there, question number 2. I hope that question is clear. If it's clear, can you just give me a thumbs up, dear children? If it's clear, yeah, okay ma'am, perfect, thank you so much. Okay, 2B, oh my god, Sanjeevani and all sitting like this. Which of these ancient men protected their body with hide and buried their dead? Which of them protected their body and buried their dead? Any idea, my dear children? Nisar Ahmed is also there. Hello. Okay, so Sanjeevani says 2B, Mansi says 2B, Abhi is there. Hello, dear. Nisar says B, and Utupal says B. Ritika says B. Okay, so B is what? Neanderthal man. Yes, my dear children. Neanderthal man were the organisms who basically started protecting their body. Okay, they started protecting their body, they covered their body, and also they buried their dead. This was very unique. Until now, no one did that. Okay, so this is very much important. Please remember, this is a very unique feature about them. Similarly, you should know the features about all of other things. Okay? Yes. Going ahead. So here I have put the features. If at all you can go through it, it is very, very, very important. Now, arrange the following animal groups in correct sequence of their occurrence on the geological time scale. Baby girl, bolo to. Krishna, the only baby girl you should be having as of now is me. Talk to me. Papa, it's exam time. You guys are talking here and there. Okay. Shreks, hello. And JNV also gave me the correct answer. Sanjini says the answer for this question is 3C. 3C. Okay, Kavita says it is C. Come on, Krishna, let's give the answer. Okay, Ritika says it is C, JNK says it's C, Shreks, hi dear, and Abhi also sees C, and Jia says C, and Udupal says C. Okay. C is what? C is this. C is this. And why do you say that this is the answer? You are being asked about what? You are being asked about which is the correct sequence of their occurrence, right? Listen to me carefully. This is the way you can remember it. From fishes, you got amphibians. From amphibians, you got reptiles. From reptiles, you got mammals. Also remember, from reptiles, you got aves as well. So aves and mammals came from reptiles only. Here, there is no aves, so you can remember like farm. And in that case, your answer for this question is going to be 2, 1, 3, 4. Okay? I hope this is clear. So Maya, C is baby girl. Correct, Baba, it's C. And yes. Ma'am, is it enough to do all the model question paper for 65 plus? No, it is not enough. Okay, you would have to go ahead, solve more questions as well. Perfect. Yes. Next question. The paleontological evidence for evolution is obtained from. From where do you get the paleontological evidence, Macha? The paleontological evidence, where do you get it from? Yeah, there are different forms of evidences. Please make sure you go through that. There is embryological evidence, there is comparative anatomy. Okay. Kavita says the answer is fossil, Samiha says fossil, Sanjeevani says fossil, Udupal, Mansi, everyone says fossil. Yes, fossil gives us what? The paleontological evidence out there. So with the help of where do we find the fossil? For example, I find it in depth. That means during that particular era where this was the topmost layer, I found this, their organisms were there. Now at the topmost layer, if I find the fossils, that means they were recent organisms. So yes, these would be definitely helping us. Okay? Next, yes. Observe the following diagrammatic representation of operation of natural selection on different traits, and natural selection depicts what? Stabilization, directional, disruptive, cannot be understood. What do you think is the answer for this particular question? Sanjeevani says the answer for this is 5C. 5C is what? Disruptive. Okay, what about others? Aesthetic softies is there. Hello, dear. Okay, Samiha and aesthetic, Sesi, Sanjeevani, Sahana, JNK, Mansi is baby girl. Neanderthal also find it the clock. That's not given in your NCERT for that. I would have to do a bit research out there. Would be wrong to say if at all I do not know it. But yes, that is an interesting question. I'll be definitely searching that. Shrek says C and Nisar has a different answer. Somya also says Somya. Is it Somnia? Yes, I believe. And Gia also C. Disruptive. Disruptive means what? Listen, you do not have to mug up over here. Understand thing. Suppose I say tall people are there, dwarf people are there. Okay. And I am saying that both tall and dwarf people are seen over here. That means it's two extremes and both of them are being selected. That is disruptive. Now when I'm saying that, okay, I do not find either tall or dwarf, basically it's the middle character. This is known as stabilizing. This is known as stabilizing, where basically there's a middle character that is being seen. Now there is one more thing out there. And that is this, where, for example, I am saying that only dwarf people are being selected. So it is going in that particular direction. That is known as directional. Okay, that is known as one particular direction it is going, it is known as directional. So your answer for this question would be disruptive. Why? Because the diagram is like this. You have two things. Okay, children, understood? If understood, just give me a thumbs up. Jeevan, ma'am, carbon paper available. PU2 board didn't give me any paper. Listen to me. It's very what I'm saying is, do not fall for every trap out there. Okay, do not fall for every trap out there, every squeaky, tricky titles out there. You follow teachers and you follow them. Huh?
What is this?
No.
Guys, maths tomorrow. Everyone, math is there tomorrow. Urgent update. I am coming live at 12 o'clock. Am I going to end before 12?
No, 12:30. Okay.
I am coming live at 12 o'clock for all the model papers. Okay? So,
Okay.
as soon as ma'am finishes, next live will be within 5 minutes.
Okay, you can do one thing. Those of you who are mathematics champions, please go ahead with Sir's live at 12.
No,
do
live
not.
will start only after your live goes. We'll do like that only. Let it be.
No, it's okay. It's okay. It's fine. It's fine. If it's important, it has to be taken, right?
We'll do after your live, ma'am. Just an update because I can see many of the math students are here. I saw Gia, okay, and many of them are maths. I think very less are there. Most of them are your fans, ma'am.
Not my fans and not
They're
my
all
students.
your fans, right?
Go.
And ma'am has paid you how much? All of you, ma'am has paid you how much? Tell me. Sahana is here. Sahana is from my class. Okay. Don't fight, sir, ma'am. No. Ma'am has paid you how much? Because I keep getting in my life also, ma'am. You're so beautiful. Okay. Why in my life, ma'am, so beautiful?
So,
Yes. See, Gia is also from
1
my
lakh,
class.
1 lakh.
Hey, ma'am has paid 1 lakh to all of you, right?
So-
I knew that something has happened.
Paid her heart. Oh my God. Thanks. Listen to.
Sahana, I'll give you my heart, but don't fall for the trap.
So listen, children, honestly, sir, if at all you want to take by 12 o'clock, you can go ahead if there is a plan like that.
Yeah, the thing is, I saw many comments in all my live chats, everything, that all of you are asking for model papers. And if I solve model papers, it will take 5-6 hours. What I'm doing is I'm going to give you a very good strategy to complete model paper within short amount of time. So you come with me in the live class. I'll give you the PDF at the same time. Okay, you'll get the solved PDF, everything. So after the ma'am's live is over,
Yeah.
after the ma'am's live is over,
When are you planning it? I am planning to end by 12:30 is what I believe.
150 questions. How? Ma'am, I
12:30.
think ma'am is enjoying too much time with you guys. She started with 8 o'clock.
No, 8:30. 8:30. Go!
Okay, fine.
No. Okay.
Okay, guys, so we'll let you know. I'll go back and you'll have a class schedule. All of you are already part of WhatsApp channel, so we'll share the schedule over there also, and we'll put the live schedule on the YouTube.
Yeah.
You can join over there for the all model papers in one video. And evening, I'm coming with that session that you are all asking. All 1, 2, 3, 4 markers means the predictable questions I'm gonna give you over there. Yes.
Okay.
No, I am heart of biology. Okay.
Listen to me, every children out there. Since sir is there, please make sure you join the WhatsApp channel for getting the updates.
Yeah.
And just go through it. If at all you're a math, if you have maths tomorrow, I told you in the video also before putting up this. If you have maths, if you have any other things to do, please do it. Okay. And also go for that particular live when it starts. So be updated regarding that.
And let everybody know I'm solving model papers if they are not aware, because this is something which is urgent, has come up from all of you, all your requests. And see, ma'am's getting angry at me.
Go.
Because I've broken her, that rhythm. Okay.
Yes, you have broken my rhythm. Go.
Thank you.
Yeah, wishing you the best. Bye.
Anyways, guys.
Okay. Okay, so I think that everyone who felt sleepy is not sleepy anymore now. And thank you for such sweet words for me, guys, Jeevana, and everyone out there. Okay, let's come back. Let's come back and let's solve it. Let's complete it. And guys, to be honest, I also want to tell the same thing itself. If at all there is anything you feel very much troubled in mathematics, and if you have math tomorrow, take it seriously. Go ahead with that. I don't mind. Okay, I am a teacher, and as a teacher, I want you guys to score good. Yes, that's all. Yes, perfect. Let's go ahead with the next question then. And over here, it goes like this. That is question number 6. Dash stage in human evolution is used hides to protect their body and buried their dead. With this, ma'am, one doubt. Yes, tell me, baby girl. Should we prepare preparatory papers too? Yeah. Preparatory papers, yeah, you can just go through it. Look, listen, any type of questions which are there, you should be practicing it. That comes from board, you should be practicing it. Whether it's from question bank, whether it's from preparatory, whether it's from model question paper, you can try out all these questions. It does not harm you in any ways. It is going to do good only. Okay? Okay. So, yes, Neanderthal, Sanjeevani says Neanderthal, Mansi says Neanderthal, Sammiha says Neanderthal, and Sommaya also says Neanderthal, and yes. So, over here, the answer for this question is Neanderthal, my dear children. Every unique part about each and every organisms out there in our, evolution of man is super important. The first human-like feature was found in what? Homo habilis. Homo habilis. The first organisms to start eating meat were Homo erectus. Okay, so all these key features out there, you need to know unique features about them. Please make sure you go through it. This is very, very important evolution of man. Yeah? Yes. Hoi, hello Prachi. Okay, question number 7 now starts the question bank questions. KCET classes when start? KCET classes are going to start right after your board examination has been done. Yes. So basically, when is math live? I think it starts at 12:30 is what I believe. But you can stay tuned, and also if I get a message, I will be updating that to you, Gia. Okay? There are 2 lives is what I believe I understood as of now. Yeah. And that was not at all staged. He actually bombarded my class. So, yes. Okay? Sanjeevani 7A. 7th A is the answer. Man has domesticated dogs and has created different breeds by artificial breeding. Have they? Yes, they have. And the next thing, statement number 2, intensive breeding of dogs signifies divergent evolution. My dear children, first thing is definitely correct. Now, intensive breeding of dogs signifies divergent evolution. We had a common ancestor here. Now they got radiated, and you have multiple species of dogs out there. You might have seen husky and everything out there, right? Do you think it's coincidence? No. This is because of the breeding program that happened, and they definitely show the, divergent evolution. So 7A, Sai also says it is A, Mansi says it is B, Kavita says it is A, Samiha says it is A. So both statement 1 and 2 are correct. I'll say the answer for this question is this. Okay? Yes. Perfect. Question number 8. Statement number 1. Antibiotic or drug resistance is due to evolution by anthropogenic actions. Anthropogenic actions means human-made actions. Now, or man-made actions. Now, statement number 2. Many antibiotic or drug-resistant organisms arise due to overusage of antibiotics. Is that so? Is that so? Check and tell me the answer, guys. When you tell the answer, can you please tell me like this? 8, A, B, C, D. It will be really good if at all you could do that. Ma'am, we have only 1 day gap for biology. I know that's going to be a bit difficult, but okay. So in that particular case, I'll request, if you're someone who have not at all touched your what do you think, your biology books, or probably are very far from the syllabus out there, there's a video that I've done. I've told initially also, there's a video that I've done where I've completed all the very important questions from all the important chapters. It's around 10 hours long. If you can, go ahead and watch it. Just trust me on that and go ahead and watch it and give your best. That's the only way that I can tell you as of now. Or probably you have to cover the 9 most important chapters out there. I have made a separate video on it. Can you please go and watch the 3 days is equals to 70 video? It will be really good. Okay? Now, Kavita says 8C is the answer. Sanjeevani says 8A and Ritika says 8C. Okay, so there is a confusion between A and C is what I believe. So listen to me carefully. Okay. Antibiotic or drug resistance is due to evolution by anthropogenic action? Yes. Why? Because we human beings take the drugs. Okay, for example, we have some bacterial infection, we took the drugs and we keep on doing that. Now, under this particular pressure, what is going to happen? The organisms which are there, the bacteria which are there, they are going to evolve. They are going to evolve in a different way. They are going to make all things different, right? So this happened because of what? Because of man-made actions. Right? Yes. And now they have become resistant to the drugs out there. So I'll say this particular statement is correct. I'll say this particular statement that you see over here is actually correct. Now statement number 2. Many antibiotic or drug resistant organisms arise due to overusage of antibiotics. Is that so? Yes. It will be definitely because of that. Because we are overusing, that is why all of this thing is happening. So I'll say both statement 1 and 2 are correct. So answer would be A. Why did you have a confusion between A and C? C is what? Statement 1 is incorrect and statement 2 is correct. No, it is also correct. This example is given for anthropogenic actions. Please make sure you go through it. Okay? Ma'am, I've scored very well, but I'm scared if the paper is tough. I've scored 68 on 70. That's really good. In MCQs, okay, I've lost marks in MCQs as a reason for watching this as well. You will score good only. It's just that it's about the concepts. Make sure you do that. Concepts have to be strong. Gia, yes, please, dear, ask me the doubt. Are you concerned about when is the live? I can just text Ankit, sir. Just a second. Yes, I have texted him, okay? I'm just waiting for his reply. Once he replies, I'll get back to you. Yes. Guys, if this question is clear, can you just give me a thumbs up? If this question is clear, can you just give me a thumbs up? We'll go on with the next question out there. Yes, thank you, Mansi and everyone. Next thing, a brown-billed duck is newly added to a farm of white-billed duck. This indicates what? A brown-billed duck is newly added. So over here, you had the white-billed one, okay, the white ones, but now I'm adding a brown one in between. What does this show? Gene flow, genetic drift, mutation, or natural selection? What do you think is the answer? Thank you, Mansi, Sudarshan, and Ritika, Sanjeevani, Soumya, Haji, and Samiha, and Akash. Mansi says the answer is D. Sai says it is B. Kavita says it is D. So I see a gap between D and B option. What do you think is correct? Think about it. Sanjeevani says it is B. Guys, think about it. Think about it. This is easy question. We cannot, okay. Ritika says D. I'll give you a clue. None of you have given me a correct answer until now. Okay, I see a correct answer now. Chandrasekhar Chavan is there and my Chandrasekhar has given me the correct answer. Says the answer is A. The answer is actually A. Listen to me carefully. This is known as gene flow. When I say that we are Indians and one person from America came into this. We have our own genes. We are brown in color, right? We are brown in color. We have our own genes for that. We have black hair and things like that. Now, we have a set of genes for that. From somewhere else, for example, an American came into here, over here, they have a different type of pigmentation all throughout in their hair, in their body, everything. The genes, they are going to carry what? Genes. New genes are going to be added up over here. Isn't it changing the frequency which was there over here? Yes. This is an example of gene flow. So here normally there were white-willed ones, that means the alleles were for that, but now from somewhere else someone came into, this is known as gene flow, not genetic drift, not mutation, not natural selection. You get it? So basically your answer for this question is gene flow. Genetic drift, my dear children, comes into action when you're talking about a random thing that occurs by chance. For example, I am saying that randomly a wind came and killed all the brown ones. What does this show? This shows genetic drift. Okay, you get it. Or I am saying that over here I have the white ones and the brown ones initially, but now somehow there are green ones also being there. How does that came? How did that come? Basically because of mutation. So you should know about the basic things out there. Answer for this is gene flow. If you have understood it, just give me a thumbs up. If you have not, ask me the doubt right away. This is important. Please know from genetic Hardy-Weinberg principle, you might get some questions. Make sure you go through it very nicely. Okay, thank you, and yes, perfect. So you got to understand it, which is why we'll go ahead. Now, change in frequency of genes and alleles is due to. Why do you think there is a change in frequency of genes and alleles? Or basically, what are the factors that affects the genetic equilibrium? That is the question. Mutation, gene flow, both A and B or neither A and B. What do you think is the answer for this? What do you think would be the answer for this? Okay. 10th answer is A. Mansi says A. Haji says C is the answer. Okay. Read the question carefully. Somaya says it is B. Sai says it is C. Sanjeevani says it is B. Okay. Guys, are we reading the options carefully? JJ ma'am, you're looking so pretty today. Thank you so much, dear. You guys look pretty always for me. Okay, so 10B. I see that most of you are not reading questions carefully. So, 2 people have given me the correct answer. Haji and Sai gave me the correct answer until now. Listen to me carefully. Over here, the answer for this question is both A and B. Both A and B. This is important. Okay, both A and B. Why both A and B? Mutation, as I said, changes the frequency out there. I told you, suppose over here you had the yellow ones and you had the white ones. So, the frequency of alleles were for these particular organism out there. Now, because of some changes out there, there are now brown ones as well. This is arising because of mutation. That means new genes are being added up, new alleles are being added up. It changes the frequency, right? It does. So obviously this is correct. Gene flow, I just explained to you, that is also correct. So both A and B will be your answer for this particular question, right? Right. So your answer is C. Did you understand it? If you have understood it, give me your thumbs up. If you have not understood it, ask me. This is the right time to do so. Okay, Samiha, Mansi and Haji, Sanjeevani. Ritika, didn't understand it? Do you need an explanation? Okay, perfect. Yes. Okay, let's go ahead. The first amphibians that lived both on land and water evolved from. The first amphibians that lived both on land as well as water evolved from. Ma'am, what is the difference between gene flow and mutation? Gene flow is basically, as I told, from somewhere else someone is coming here, or probably from here a group of organisms, some people are going out. It increases the frequency, it decreases or basically increases the frequency of different alleles out there, right? So for example, you just think like this. I have a region, I have tall people, I have dwarf people. In this region, only tall and dwarf people. And all of a sudden, I have some other, medium-heighted people coming from there to here. I have a new allele being missed. This is known as gene flow. Ma'am, my name, my name is Jasmine. Yes. Okay. Okay, D. Okay, D is what? D is froga. Frog is the answer. Liston says it is B or C. Okay, Sanjeevani says the answer for this question is C, and Mansi also says it is C. My dear children who have given me any of the other answer, please understand it is not correct. And Jia, I have got your answer. The live starts at 12 o'clock. Okay, the live for mathematics for model question paper starts at 12. Okay, so listen. The answer is law of fin. Why? So I've already told you fishes, amphibians, reptiles, mammals. From here, aves also, that is the birds, right? Farm, that is how you do it. Now, amphibians are the ones that live both on land and water, and fishes came before that. And you have been asking, what the first amphibian that lived both on land and water evolved from what? Evolved from what? Fishes. But not exactly all the fishes turned into amphibians. There's a certain group of them. What do you call them? You call them as lob fin. Why do you call them lob fin? They were like fishes, but their lobes, my dear children, were very thick in a way that they could basically walk with those. So they were in the water, but they could paddle up like this, and they could come on the land. So that is the link that we have between fishes and the amphibians. So your answer for this is known as lob fins. You also have another name for it, and that is coelacanth. Okay, coelacanth is also the other name for it. Please make sure you got through this. Ma'am, lob isn't a fish, it's a fish. It's a fish, but again, I'm telling you, it's like a connecting link. So not all fishes are like this. Right, not all fishes are like this. So it's like a connector between fishes and amphibians. They come under fishes itself. Ma'am, what's your name? I told my name, dear. My name is Jasmine. Okay? Fine? Yes. Guys, Sagar, is there anything that I can help you with? Everyone else pay attention. This time is precious, right? Let's go ahead. I hope this concept is clear, and we'll go ahead. It's like crocodile, ma'am. It's not like crocodile. It's just that a normal fish, but it has a fin that is very thick, such that, you know, you have seen a normal fish, right? Do you think it has a very strong fin? No, it does not have a strong fin. But here the lob fin had a strong fin, which is why it could move around. Okay? Yes. Sahana says, oh, Aladdin wala Jasmine. Yes, you know what? My brother actually put up my name because he watched the cartoon Aladdin that time. Yes. I hope he didn't watch the other cartoons, and by chance I was saved from other cartoonish names are there. But yes. Okay, next question that we have, the biggest, okay, by the way, also one more very important thing, there is a link between reptiles and mammals also. You call them shrews. You call them shrews. Between reptiles and mammals, you have shrews. Please remember that as well. Very, very, very important. Okay, the biggest land reptiles that existed during Mesozoic era was... The biggest land reptiles that existed during Mesozoic era was Ichthyosaurus, Tyrannosaurus, sea cows, and or basically crocodiles. Ma'am, if lob fins isn't given in the option, only fish is given, then you go with fish. Okay. Sanjeevani says the answer for this question is B. Mansi says it is B. Liston says it is D. Sorry, B. Sorry, my bad. Okay, Liston Diziza says it is B. What about others? Sai also says it is B. B is what? Tyrannosaurus. Exactly. Tyrannosaurus rex was the biggest land reptile which has existed during Mesozoic era. So your answer for this question is Tyrannosaurus rex. Please make sure you remember that. Very, very important thing. Okay? Yes. Sahana, no. C is not the answer. It is B. Now, going ahead with the next question, statement number 1. Hugo de Vries believed mutation leads to spontaneous evolution. Hugo de Vries said that mutation, it leads to spontaneous evolution. Okay, and statement number 2, he said that mutations are random and directionless and causes saltation. Salted, what do you call, mutations are random and directionless and causes saltation. Sam, can we come talk about this over here? Can you try solving the questions? Ma'am, 12th question, please. This one, the biggest land reptile, it's just something that you have to remember as such. There is no, what do you call, explanation around it. It is Tyrannosaurus rex. It is a type of reptile which was there, and it was dominant during Mesozoic era. So straight question, dear. Yes. Now, if we come over here, I see that 13. Supriya is saying the answer for the 13th is B. And rest everyone is saying it is A. Soumya and Liston and Asha and Sahana, Chandrasekhar, everyone says it is A. Okay. Is it A? Both statement 1 and 2 are correct. Yes. Listen to me carefully. Hugo de Vries and Darwin had two theories. Okay. Darwin said that yes, variations happen, and variations happen because of that you have evolution that is taking place. But according to him, he said that it is very gradual process, a very slow process. The term gradual is important over here. Okay, that is a very slow process. Now on the other hand, Hugo de Vries came and said that mutations happens, and mutations are going to be very fast. It leads to spontaneous evolution. Does it? Yes, he said that, and that is correct. Next thing, statement number 2. Mutations are random and directionless and causes saltation. Do you think it's random? Definitely it's random. It happens by chances. It can be good, it can be bad. And does it cause saltation? Saltation means single step speciation, as in because of one mutation out there, a whole new species have been formed. Do you think that can happen? Yes. So both of the statements are actually correct, and this is according to Hugo de Vries' theory. According to Darwin's theory, everything is going to go very, very slow. Okay? So your answer for this question, saltation has single step large mutation. Yes, single step large mutation that leads to what? Speciation, a formation of new species. Very important thing. Ma'am, didn't understand the second statement. I can make you understand it. So listen. Okay. Mutations are random and directionless. That means, for example, over here in my DNA, suppose when I'm talking, mutations are happening. Does it occur because it is planned? No. It can be occurring any single time in my body. Okay, and directionless means it can be like, it is not always for good, it is not always for bad. It can be in either of them. We do not know about that. And the next thing, it causes saltation. Saltation means sometimes a single mutation which is there, it can give rise to a whole new species. For example, human beings are there, but there are some mutations with me which makes me a whole different species. Can that happen? Yes, that can also happen. Okay, it can also happen. So please remember that. Okay? Sagar. Okay, perfect. So Priya, will I get 20 on 20 with this? You can definitely try your best. I am trying my best to cover all the important concepts out there, dear. Okay? Yes. Sudhanva. Okay. Hi, Sudhanva. I'll try to remember that name. Okay? So, okay. Elongation of neck in giraffes due to inheritance of acquired character was given by. Elongation of neck in giraffe due to inheritance of acquired character was given by what? Sanjeevani says the answer for this question is A. Okay. What about others? Supriya, most welcome, champ. Liston says it is C. Okay. One answer for A, one answer for C. Elongation for the neck in giraffe. So he said that, whoever said that, he said that basically giraffes have longer necks because they stretched up like this. The smaller ones, they stretched up like this, and since it happened like that, it got acquired. Every single organism that came from them, they acquired it. That's what they said. Ma'am, C. Soumya says C, Mansi says A. So C and A is where you have the confusion. The answer is A. The answer is A, that is Lamarck. Okay, elongation of neck in giraffe is due to inheritance of acquired characters was given by Lamarck. And it was said that it was a big blunder. Why? Listen to me carefully. So he said that basically giraffe has longer necks. Why? Because these guys, my dear children, they were smaller ones, but they elongated their neck like this because the food was at top height. That's why. And that got acquired. That's what Lamarck said. So Darwin came into the picture and said, how can that happen? Obviously, that is not true. Since acquired traits are something that will not be going ahead. If it is there in our genetics, it might go ahead, but something that we acquire, we cannot go ahead. For example, they did an experiment, not Darwin, someone else did an experiment where they took a mice, and over here they cut the tail of the mice. And now what do you expect? The babies that this particular mice will produce should also not be having the tail, right? But do you think that will happen? Obviously, they will be having their tail. Because this is an acquired character that you acquired during your lifetime. This is not an inherited character. That is why Lamarck's principle were being disapproved. So if we stretch our neck, will be giraffe? I am short. Even I am short. Definitely that does not work. Tried it out. Didn't work out. It has to be there in the genetics. So yes. So yes, your answer for this question is A. I hope you'll not forget it. Everyone understood? If understood, can you just give me a quick thumbs up and then we'll go ahead with the next thing. Yes, sir, will be classing at 12. Starting at 12. Okay, next thing. Let's go ahead with the next thing. Single step large mutation leading to speciation is also called as. Single step large mutation that leads to speciation is also known as what? Chandrasekhar A. Liston says D. Okay. Tippaswamy Shetty says D. Chandrasekhar A. Heart for you, thank you. Sanjeevani says D and Sai says D. Okay. Arun, there will be a math session right now at 12 o'clock. It has started. Go watch that live. Okay. And Sudhanva says D, 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 D. Supriya, Sahana, Liston, Haji, Sahana. Everyone says D. D is what? Saltation. And this is actually correct. So which is why I'll go ahead with this. Perfect. You guys got it. Understood it. Now the organic compounds formed in Miller's experiment are, you know Stanley Miller's experiment. This is one of the most important thing out there. And in Stanley Miller's experiment, my dear children, he saw that some things were being formed, okay, formed in the experimental tube. What was that? 16th question answer. 16th question answer. Guys, if at all you answer me like this, it will be really easy. 16th A, B, C, D. Okay, Tippaswamy says it is A, Sanjeevani says it is A. Okay, A is what? Amino acid only. Sahana says it is A. Okay, Sai says it is B, Liston says it is D, Ayu is there, hello Ayu, Ritika says it is A, Mansi A, Sudhanva says A. The answer for this question is A only. Listen to me, even though in your NCERT multiple things are being written that this is formed, that is formed, that is formed and things like that. But they have clearly mentioned in S.L. Miller's experiment only amino acids were formed. Nitrogenous bases, pigments, fats and everything were formed by other experiments done by other people, not by them. So please understand the answer over here is amino acids. Do not forget it. At night 12 a.m. also, if at all JJ ma'am comes and asks you, you should be able to answer this. Okay? Deeksha is there with the name Deeksha. A. Okay, okay. Hello, my name is Rakshita ma'am. This is my father's account. Okay, I'll definitely try to call out that particular name itself. Yes. Okay, let's go ahead. 17th question. Embryological evidence for evolution was disapproved by. He finally obtained that from that experiment was amino acid. Yes, exactly. So embryological evidence for evolution was disapproved by Ernst Haeckel, Carl Ernst von Baer, and Charles Darwin and Alfred Wallace. What do you think is the answer for this? So Dhanva says it is B, and Tippaswamy Shetty says it is B. Ritika says it is B, and Rakshita says it is D. Okay. Guttu, hello ma'am, I'm from Guttur. Hello dear. Prashant also says B. Deeksha says it is B. Is the answer B for this particular question? Carl Ernst von Baer, is that so? Disapproved by this guy? Yes, it was disapproved by this guy. I hope you guys know what do you mean by that. Listen to me. Embryological evidence basically means we have studied that farm is there. That farm is there, fishes, amphibians, reptiles and mammals, right? Right. This is something that we know that. Now listen to me over here. When I say that we have evolved, how do we prove that? Because we are seeing that when we human beings, you, me, Ritika, Guttu, Haji Ali and Rakshita, Ayu, everyone, Sahana, everyone out there, when we were in our mother's womb, we had gill slits. Do we have it now? We had tail. Do we have it now? Everyone look around. Do we have tail? No. What does it show? Probably we came from these organisms, which is why we have all of this in our embryological stage. And that is the embryological evidence as well. Okay. But then that was given by Ernst Haeckel. That was given by Ernst Haeckel. But another Ernst guy disapproved it. Yes, ma'am, we have, ma'am. Okay. Another guy disapproved it. That was Carl Ernst von Baer. He said that in that case, you should be having some features of reptiles also. You should be having some features of amphibians also. That is not something that you are having, right? You just have some features of fishes and that's it. How can I say that? That obviously does not work like that. So, my dear children, your answer for this question is Carl Ernst von Baer. He was the one who disapproved this. The last bone of our spinal cord. Exactly. In that way, yes. Yes, the tailbone. Okay. Now, next thing that we have is question number 18, that is, arrange the following plants as appeared on different geological periods. Now, I see, hello Mohi, hello Mohi. I hope you're doing great. By the way, mathematics live has started. If at all there is any math children over here who really want to study maths, please go ahead with that. Yes. Ma'am, it's correct, na? Yes, it is. We have actually, yeah. And you know, some people are born with tail also. You might have seen those conditions. Yes, let's go. Tippaswamy says 18th answer is A, and Rakshita says it is B. I think the previous question or this question. Okay, and Ritika says it is A. Sanjeevani is still here and says it is A. Dear, if you have mathematics, you can go ahead, okay? I do not mind at all. And ma'am, when will be our next class? Tomorrow at 4 o'clock afternoon, you will be having complete biology. Ayu says it is A. A is what? Conifers, needles, dicots, monocots. For those of you who gave me the answer for this, I believe that you guys are awesome, because it is difficult to remember it. That's why I'm telling you. So the answer for this question is actually A. Conifers came first, then needles, then you have dicots, then you have monocots. So how do you remember that? Listen to me carefully. Here, C square comes first. Both of these were gymnosperms that were there. Okay, these were also gymnosperms, but these C square came first in the alphabetical order, so C comes first, right? So C came first, then you have ginkgos and needles. These are gymnosperms. Now from gymnosperms, my dear children, you got angiosperms. In angiosperms, who came first? Dicots came first. And then came what? Monocots. Then came monocots. So always remember that, know the flow. Apart from that, you have ferns and spinosad. They are basically pteridophytes. So pteridophytes, first came algae, then came bryophytes, then came pteridophytes, then came gymnosperms. From gymnosperms, basically you got angiosperms as well. Know the era. This is during Mesozoic era. In Mesozoic era, especially during Jurassic and Cretaceous, all things were happening. So please go through it. It will be really, really good. I said B. It's okay, dear. Deeksha says A. Ma'am, when will the session end? I am thinking to end it by 12:30 whatsoever. But yes, if at all you guys stay, if you have just biology, then you can stay longer, and I'll take. I do not have any problem. Okay, I'm here for you guys. Yes, perfect. So let's go ahead. 19th question. Replacement of the lighter colored variety of peppered moth, Biston betullaria, to its darker variety of moth in England is an example of. Is an example of. What do you think is the answer for this particular question? Very, very, very important. Replacement of lighter colored variety of peppered moth to its darker variety in England is an example of. Bye, ma'am. I am. Okay. Bye, ma'am. Going for math session. Nice to meet you. Thank you for teaching by your lovely heart. That's so sweet of you, Ritika. Please take care of yourself. Wishing you the best for mathematics. And yes. Okay, Sanjeevani says 19th is A. Tippaswamy says it is A. Is baby girl says A. Sanjeevani A. Haji says A. Ananda also A. Okay, so over here, A is what? Natural selection. Was it natural selection? Yes, the nature selected it. Listen to me very carefully. Initially, you had what? You had plain, what do you call, trees out there, which were covered by lichens. Lichens, okay? And these lichens which are there, they are not found in areas which have too much amount of pollution. So these guys were there. And you had white moth and you had black moth. These things were white in nature, which is why the white moth, whenever it sat over here, it was not spotted by any of the other organisms out there. But into this white background, if at all I put something black, don't you think it will be visible? Obviously, yes. These guys were eaten up. So the Biston, what do you call, Biston betullaria, which was there, it was the dominant one first, okay? And Biston carbonarae, which was there, the dark one, it was recessive because it was not able to show its function or that it was being easily spotted. After that, my dear children, after some point of time when pollution and everything increased so much, the lichens were not at all found. Lichens, remember, they are natural pollution indicators. They will not be found when you have too much amount of pollution. Now in this particular scenario, your background is black. Now the black ones which were there, they will survive, but the white ones, if they sit over here, I can go pick up them and eat them up. Remember, this is very important. This is natural selection. Okay, ma'am, can you teach maths? You're so sweet. Oh my God. If I go around teaching mathematics, probably you will hate mathematics from that day onwards. Ma'am, do you teach for NEET? Yes, but over here in this channel, we have been taking a break for NEET for some time now. But yes, by camouflaging. Exactly. Children, are you understanding? If yes, can you just quickly give me a thumbs up? Can you quickly give me a thumbs up if at all you're understanding? It will be really good if you do so. Yes? Perfect. Thank you. Okay. Next question. At a particular locus, frequency of allele capital A is 0.6. Frequency of allele capital A is 0.6, okay? And frequency of allele small a is 0.4. Fine. What would be the frequency of heterozygotes in a random meeting population at equilibrium? My dear children, what do you think is the answer for this? I love biology because of you. That's so sweet of you. Thank you, Haji. And over here, guys, try out this particular question. Guys, listen to me. Suppose it is capital A, capital A. What do you call this? Homozygous condition, right? It is small a, similarly, homozygous condition. Suppose it is heterozygotes. How do you say? It should be capital A, small a. That is what you mean by heterozygotes. You have to find the frequency of these type of organism who would be having an allelic combination like this. How will you be able to find that out? Listen, that is a formula that you need to remember. Okay, so 20, Tippaswamy says it is D, Haji says it is C, Sanjeevani says it is C, and yes. So listen, let's try to follow it. Let's try to do it by ourselves. Deeksha says it is D. Okay, let's try to solve it. There is a formula that is P plus Q whole square is equals to 1. If I elaborate it, P square plus 2PQ plus Q square is equals to 1. What is P square over here? That means capital A, capital A. What is Q square over here? That means if we want to find the frequency of recessive homozygous thing. Now over here, 2PQ is something that is there for heterozygotes. So this is both for homozygotes, this is for heterozygotes. 2 is there, P stands for 0.6 and Q stands for 0.4. What is your answer? Your answer comes out to be 0.48. Some of us forget about this 2 part. Make sure you do not forget about it. The answer for this question is going to be D. It is going to be D. Okay, very important. You are being asked what is the frequency of organisms who have homozygous dominant allele. How do you find that? You have the homozygous dominant allele 0.6. You do A square or basically P square. That is 0.6 whole square. Your answer will be 0.36. That is how you find it out. Okay, this is super important. Make sure you know the concept really well. It is going to help you out. Okay. Deeksha, Ayu and Sai, and before that I think Tippaswamy and yes, got me the correct answer. So if at all it's clear, just give me a thumbs up. If it's clear, can you just give me a thumbs up? We'll go ahead with the next question. Yes, perfect. Let's go ahead. Next, statement number 1. Pouched mammals. This might be a bit difficult question. This is directly from your question bank and it's difficult. So make sure you listen to very carefully. Statement number 1. Pouched mammals of Australia survived because of lack of competition. Statement number 2. Continental drift has both positive and negative effect on population. Now those of you who have read the NCERT like literally line by line would be able to do this answer. If not, you might find a bit trouble, but do not be worried. I've got you covered. I give you the answer also if you do not know the answer. But you need to understand by this is that you should not be just scared. Not every question is going to be like this. Okay? Ma'am, you're teaching very nicely. Thank you so much, Deeksha. Ma'am, I asked you to teach maths and that is what I meant. Okay. So sweet. Yes. Children, come on. Come on. Let's try solving this particular question. By the way, everyone who is watching this, new or old, I hope you guys have subscribed to the channel. Do that and solve this particular question. I'll tell you where is it found. If at all you want to read your NCERT and give me the answer, I do not mind it. It's found after this particular thing. That is evolution of animal is given, right? In the reptile portion, reptile to mammal portion, you will be able to find this in the paragraph, in that particular paragraph. Haji says the answer for this is A. Manju, hi dear. Sai says the answer for this is B. So, let's see. Pouched mammals of Australia survived because of lack of competition. This is actually correct. Continental drifts have both positive and negative impact on the population. That is also correct. So, what is given in your NCERT? Listen to me carefully. This is exactly given. Okay? When reptiles came down, mammals took over the earth. Reptiles, when they came down, mammals, they took over the earth. Done. There were South American mammals resembling horse, hippopotamus, a bear, rabbit, etc. So there were what? South American mammals who were resembling all these organisms out there. Due to continental drift, that means the continent got apart, my dear children, there was a drift out there. When South America joined North America, so when South joined the North America now, the animals were overridden by North American fauna. So South all were living very nicely, but because of continental drift now, South joined with North, okay. But now when they came there, northern fauna, that means northern animals overridden them. That means basically they were the dominant ones, the north ones, okay. Now, due to the same continental drift, pouched mammals of Australia survived because of lack of competition. But on the other hand, these guys were getting harmed. But on the other hand, there were some pouched mammals of Australia who did not have any competition. Why? Because of continental drift, all the competitors went away. You get it? So, continental drift is good and bad? Yes, it is. Pouched mammals of Australia, did they survive because of lack of competition? Yes, they did. This is a statement from your NCERT, and these type of questions can be expected, but not too many. Okay, so please know about this also. I hope it had helped you. If you understood it, please give me a thumbs up. If you have a question, please ask me. If it's all good, we'll go ahead. Perfect. Next chapter, that is microbes in human welfare, and let's go. Awesome. Okay. So let me see how many questions are there in this particular chapter, if not too many. Okay. Okay. Question number 1. Saccharomyces cerevisiae, commonly called brewer's yeast, is used for fermenting what? Is used for fermenting what? Deeksha, ma'am, two marker, teach in easy way. Dear, I have done a separate video on 2 marker. If at all you're very free, please can you watch it? It will be really, really helpful. Okay, Sanjeevani says 1B, Liston says B, Tippaswamy says B, and Rakshita says B, and Sai says C. Haji says B. Okay, so most of the answers are B only. The answer for this question is also B. Please understand this. Why B? This is yeast, Saccharomyces cerevisiae, and they are there for fermenting malted cereals. Malted as in something that was about to do germination, but now they have stopped. Okay, why were they about to do germination? Why were they given condition to germination? In germination, my dear children, the complex carbohydrates are broken down to simpler ones, right? And for fermentation process, you need the simpler ones only. So if at all you give conditions for germination, this process have halfway done, and now the fermentation can easily happen. So you use fermented malted cereals, you use for fermentation malted cereals and also fruit juices. Very important thing, please make sure you know that. Okay, so answer for this question is definitely going to be B. Next question. Cyanobacteria is an autotrophic microbe widely distributed in aquatic and terrestrial habitats. Can fix atmospheric nitrogen is the cyanobacteria, which is widely distributed and can also fix atmospheric nitrogen is what? The answer for this question would be nostoc. Nostoc is the cyanobacteria. Please understand, bacteria, cyanobacteria are 2 different things. They come under 1 category only. Plain bacteria do not make their own food. Cyanobacteria prepare their own food. Please remember. Both of them can fix nitrogen, but cyanobacteria can prepare their own food, which is why they are put differently. You always remember no. Instead of you know, no. Anabena, nostoc, oscillatoria, they come under cyanobacteria. Rhizobium, acetobacter, azotobacter and azospirillum come under bacteria. Basic differences. All of you were correct. Sanjeevani, Mansi, Tippaswamy, and Sai, and Haji, and Sahana. Perfect. Now, question number 3. Because of the presence of which of the following vitamin, curd is considered as nutritionally richer than milk? Okay, because of which of the following component, curd is considered to be nutritionally richer than milk? Vitamin B1, vitamin B2, vitamin B9, vitamin B12. What do you think is the answer for this? Sahana says, okay, wait. Sanjeevani says it's 3D, okay. And Mansi says D, Tippaswamy says D, Haji, Sahana, Manju, and Ayu, Sai, oh my god, Gadaya. I'm not able to pronounce your name, but yes. So answer for over here is this. Agad, are you Agad by any chance? Varalakshmi is also there. Everyone is saying D is the answer, D is the answer. So when the lactic acid bacteria convert the milk to curd, they release vitamin B12, which is why the curd will also be having vitamin B12. Okay. Mycorrhiza is involved in the absorption of dash from the soil and passes it to the plant. Mycorrhiza is involved in the absorption of what from the soil? Any idea? Deeksha also gave me the correct answer. Sanjeevani says 4B. 4B. Phosphorus. Mycorrhiza is what, Baba? Mycorrhiza is a combination of a fungus and the plant roots. So plant root is here, the fungus is here. The fungus can go ahead deep into the soil and trap the phosphorus. This is one very important feature of mycorrhiza. Okay, and the fungus belongs to what? The fungus over here belongs to glomus. Please remember it. They belong to glomus. This is also super important. So your answer for this question is going to be B. Okay, it is going to be B. Sanjeevani, Tippaswamy, as well as Haji, Mansi, Sai, Dagat, and Ayu, everyone are correct. Next question. During sewage treatment, the gas produced in anaerobic sludge digester includes anaerobic sludge digester. You're talking about anaerobic sludge digester. What is the gas that is being produced? Please understand, when you talk about anaerobes, that means the ones that do not need oxygen. And when you talk about the gas, always understand it is going to be biogas. Because biogas are produced by anaerobic bacteria out there. Right? So biogas is made up of what, Macha? Sanjeevani says the answer for this question is C. Haji says it is B. Sai says it is C. And what about others? 4B. This is 5B. Okay, sorry. Deeksha says C. Okay. The answer for this question is C. Remember, biogas is not just made up of methane, but of three things. Methane, and then you have hydrogen sulfide, and then you have carbon dioxide. Do not go and write ammonia and come, okay? Methane, H2S, and carbon dioxide. These are the three things which makes up this. Sometimes instead of H2S, they can also give hydrogen. If at all in your options, none of the options you have H2S, rather you have just H2, you can go with that also. I do not mind, nor does the PU2 mind. Okay, perfect. Yes. Next thing, question number 6. Dash is the producer of butyric acid. Who produces butyric acid? Any idea, my dear children? Which organism is the one that produces butyric acid? Clostridium butylicum. Sanjeevani says Clostridium. Ma'am, if they give both H2 and H2S, H2S. Then go H2S. Okay, Clostridium butylicum. Mansi says butylicum. Which option to choose? Okay, Clostridium butylicum. Your answer is Clostridium butylicum. It is the one that gives rise to butyric acid. Please understand, this is a bacteria. So please know all these organisms which are given, are they bacteria, are they fungi? That is also important. So Tippaswamy, everyone, Haji, and everyone out there who gave me the correct answer, thank you. Over here, I have a list of the things which are produced. Citric acid by Aspergillus niger, it's a fungus. Okay, acetic by Acetobacter aceti, that is easy to remember. Butyric acid, Clostridium butylicum. Lactic acid, Lactobacillus, three of them bacteria. Ethanol, in fermentation what do you get? You get ethanol, right? So ethanol is produced by Saccharomyces cerevisiae, yeast, and yeast is also a fungus. So these two guys are fungi. Rest everyone is bacteria. Please make sure you know about that. Okay. 7th question. Now this is from the question bank, so probably we need a bit of efforts up there. Okay, Propionibacterium shermani is used in the production of. Propionibacterium shermani is used in the production of. Sanjeevani says 7B, Mansi says 7B. Okay, what about others? Agad says B, and Haji says B, Tippaswamy B, Sai says B, Manju says C. Okay, see multiple answers out there. The answer for this question is going to be B, Swiss cheese. Have you seen Tom and Jerry in that? The Jerry always looks out for cheese with big holes. Why there are big holes out there in the Swiss cheese? Because the Propionibacterium shermani makes up it and it releases so much amount of carbon dioxide, and the way through which it releases, there will be some big pores out there. Okay, so your answer over here would be Swiss cheese. By the way, any idea about which organism produces Roquefort cheese? You do not have to tell the name of the organism, you just tell me whether it's a bacteria or a fungi. Roquefort cheese which is there, or Roquefort cheese which is there, which organism produces it? A bacteria or a fungi? Fungi, exactly. Remember that, okay? It's a fungi that produces it. Here it's a bacteria. This guy is a bacteria. Perfect. Next thing, Alexander Fleming discovered penicillin while working on. Alexander Fleming, who is there, he discovered penicillin while working on what? Any idea? Mansi says it is, okay. Sippaswamy says it is A. Manjeevani says it is A. Sai, I am born and brought up in Indore, Madhya Pradesh, but my roots are from Kerala. Okay. Haji says it is B, and B. What do you think is the answer for this question? Sanjeevani and Mansi are the only ones, I think, who gave me the correct answer, and also Sippaswamy gave me the correct answer. It is A, Baba. It is not Streptococcus. Most of us get confused with this and this. Do not get confused. Remember, Alexander Fleming has LL so many times. Here in Staphylococci also it has L. I definitely understand why there's a confusion, because the names are quite similar. But it is Staphylococci. He was working on Staphylococci. One day he forgot to wash the dish. The next day he thought that, oh my God, it is going to be spread up. But then to his surprise, he found that the bacteria was not able to grow. Why? Because a mold was developing over here. What was the name of the mold? Penicillium notatum. Penicillium notatum. What did this mold do such that the bacteria was not able to grow? They released some antibiotics. What is the name of this antibiotic? Penicillin. Penicillin. Ma'am, is this a bacteria that is producing antibiotic? No, it's a fungi that is doing that. Penicillium notatum is a fungus that is doing that. Staphylococci is a bacteria right over here. Okay, so you can remember like LL and it will be easy for you to understand. This is the thing. Got it? Everyone who understood it, just give me a thumbs up, and then we'll go ahead with the next question out there. Yes. It's also called as water drug. Is it? Okay. Perfect. Which one of the following alcoholic drinks is produced without distillation? Without distillation. Any idea? What do you mean by distillation first of all? Distillation means basically it's undergoing a process where all the water content is going to be evaporated. That means whatever is left behind is going to be more concentrated, right? So the things that undergoes distillation will be having more concentration of alcohol. The things that does not undergo distillation will be having less, I mean, it's because it is diluted. It's not having less content, but basically it is going to be diluted, not concentrated. So, yes. Thank you so much, Haji. Okay, so Sanjini says 9A is the answer. Tippaswamy says it is A. And Swansi says it is A. Sahana A. Haji Ali says A. Okay, wine and beer is the answer. So listen to me very carefully. The answer is definitely wine and beer. Without distillation means obviously the alcohol content should be less. And in wine and beer, so many people drink it, right? Wine and all. So it definitely has less amount of alcohol in it. Whiskey, rum, brandy has too much concentration of alcohol in it, which is why I'll say it is produced with the help of distillation rather than others. Yeah? No, dear Sai, I do not know it. Yes. Next question that we have is question number 10 that says, activated slug in the sedimented is the sediment deposited in which of the tanks? Activated sludge is the sediment deposited in which of the tanks? Any idea? Activated sludge is the sediment deposited in which of the tanks? Settling tank, sedimentation tank, aerobic tank, and aerobic sludge digester. What do you think is the answer for this particular question? Sanjeevani says it is 10D, Mansi says it is 10A, Richie says it is A, okay. Tippaswamy says C. Guys, if at all you can give me the answer like 10, 10, 10, it will be really good if at all you put the number. Okay, A is what? A is the settling tank. Is it the settling tank? Yes, it is the settling tank. Remember the first process, the primary filtration and the sedimentation is done in a settling tank. Then you take everything to the next thing, that is an aeration tank. Then again, the flocks and everything, you put it back into the settling tank. And finally, you use anaerobic sludge digester. So, at the third place is where basically the activated sludge is being formed. This is nothing but the flocks and everything. So, it is the settling tank. Your answer is going to be A. Okay, your answer is going to be A. Madam, Nyan Lalatin fan ano? How can I say that you are Lalatin fan? Okay. Got it? Saya. Okay. Now this question, guys, make sure you pay attention to it. Biogas is a mixture of what? biogas is a mixture of what? CH4CO2 H2, CH4SO2 CO2, CH4NO2 H2 or CH4SO2 H2. Tippaswamy, okay, before that, Sanjeevani gave 11A, Tippaswamy got A, Mansi got A, Sai also got A. So answer is A. CH4 as in methane, CO2 as in carbon dioxide, and H2. As I already told you, it can be H2 or H2S. Okay, so it is A. Next thing, match column 1 with column 2 and select the correct option using the codes given below. So using the codes which are given below, you have to choose the correct option which is out there from here. Any idea? Twelfth question. Sanjeevani says 12th answer is B. 12th answer is B. Citric acid is produced by what, my dear children? Whenever you get matched the following question, first of all, you have to read all the options, then start solving the question, okay? So citric acid which is there, it is the one that is produced by Aspergillus niger. A goes with 3. A goes with 3. How many options do I have? I just have 2 options like that, which is why I'll just cut. I have 3 options like that, which is why I'll cut the option which does not have it, and that is this one. Okay, the next thing that I have is go with the last option, butyric acid. It is formed by Clostridium botulicum. D goes with 2. I just have 2 options with that. 2 options like that, so I'll cut this option also. Now I have just left with those 2. Cyclosporin A, my dear children, is produced by what? Trichoderma polysporum. Okay, cyclosporin A is what? Immunosuppressive agent, if at all you remember. And lastly, you have statin. Statin is produced by Monascus purpureus. So over here, your answer for this question is going to be what? It is going to be B. Cool? Yes. Perfect. Next one. Which of the following is not an advantage provided by mycorrhiza to the plants? Hi, Vinayak. Which of the following is not an advantage given provided by mycorrhiza to the plants? What do you think is the answer for this particular question? 13th D, Sanjeevani says 13th D, Mansi says 13th D, Tippaswamy says B, and Sai also says B, Sanjeevani says B. Okay, Richard Anthony says D. Let me see, what is the answer? It is going to be B. Why B? Let's listen. Resistance to root-borne pathogens. Yes, it provides it. Tolerance to salinity and down. Yes, it provides it. Overall increase in the plant growth and development. Yes, it provides it. And also it helps in phosphorus absorption. But increase in the yield quality, it does not provide it. This is something that is out of the box, which is why I'll say that this is going to be wrong. This particular statement is going to be wrong. Got it? Super, super, super important thing. Fine? Yes. Now, next thing. I hope, children, you have got it. If yes, can you just give me a thumbs up? If yes, can you just give me a thumbs up if you have understood it well? Oh my God. 14th A, Sanjeevani says, okay, thank you. 14th A, and Sanjeevani also says 14th A, and Haji also says 14th A. The microbial biocontrol agent for butterfly caterpillar is Bacillus thuringiensis. Why? Basically, it produces toxin, protoxin or prototoxin, and inside the body of the caterpillar, it becomes active. Why? Because it has an alkaline pH out there, right? So the biocontrol agent for butterfly caterpillar especially is Bacillus thuringiensis. It's a very easy and straightforward thing. Sai, Vinayak, and Timpe Swami, everyone gave me the correct answer. Mansi also. Yes. And over here, I have all the things been written. Make sure you go through them. Very, what do you call? High chances of them coming up there. Which of the following is a bacterial biofertilizer? Now here I have to check your knowledge. Okay. Yes, Hadi. I'll be definitely doing that. Okay. Guys, come on. Which of the following is a bacterial biofertilizer? Which of the following is a bacterial biofertilizer? Tippaswamy says, okay, 15C, Sanjeevani says C, and Mansi also says 15C, Vinayak says C, and Rakshita is also saying C, and Sai says C, Haji also says C. C is Rhizobium, Is it rhizobium, which is a bacterial fertilizer? Yes, because no, everything else is cyanobacteria. If you're talking about proper bacterial fertilizer, it has to be rhizobium or oscillatoria or, sorry, not oscillatoria, I mean azospirillum or acetobacter. So you guys are awesome, you have understood it. Super proud. Yes, perfect. Now, my dear children, I would like to stop right over here and ask you one thing very important. It's already 12:34, and with what I promised, I said that I'll be going until 12:30 for you guys. Now, I understand that some of you have mathematics examination, which is exactly why I'm thinking of stopping this particular live. There are a few more chapters, that is biotechnology, principles and processes, biotechnology and its applications, and what else is there? Organisms and population, biodiversity and conservation, and you have ecosystem as well. That means 5. A total of 5 chapters are still left. Yes, I'll request you one very important thing right over here, and that is I'll show you a video where you can go ahead and find the rest of the answers, or basically the rest of the chapters out there, since I really do not want you guys to stress up on anything. So I'll just show you. Listen to me carefully, okay? Do not go anywhere. I'll be guiding you with something. Yeah. Okay. So basically over here, you see there is a playlist. Okay, there is a playlist been given. I can just reduce the lighting a bit such that it is visible for you. Okay. Yes. So there is a playlist right over here. If you click on that particular playlist, you would be seeing something like this. Second PUC Biology Final Revision. Okay, second PUC Biology Final Revision. What you have to do is, you click on the view full playlist out there. Now in this particular playlist, you see top 50 questions, all MCQs. So it is a 3 hour 39 minutes. So same particular questions that I did in 3 hour 39 minutes itself, because there is no talking involved, right? So please understand that you can go ahead and watch this video. For the rest of the chapters which are there, exact questions will be there. It will be covered over here. Please make sure you watch it. Do not miss it out by any chance, especially for the last chapters out there. It can give you confusing questions. I do not want you guys to miss out on it. Now 2-3 markers, it's right over here. Okay, and there is what do you call, complete biology. Now this video is for everyone who feels that I have no idea what to do in biology. Okay, there is literally no idea of, I might even fail the examination. If at all you have a sense like that, and if you do not understand where to start, where to stop, there is a video like this. It's complete biology, 1 marker, 2 marker, 3 marker, 5 marker, everything is being merged over here and given to you. The timing is 10 hours 18 minutes. It's all recorded video. There is no bug in between. If you watch that, I can guarantee you, you understand the concepts. It will be much more productive than doing chapter wise, what do you call, going chapter by chapter reading NCERT. I'll recommend you can go ahead and solve more questions, obviously. But if you feel, this is only for the children who feel I am destroyed, I cannot do anything. Rest of you, please go ahead with your own loop itself. I was just giving you resources. It should not be like you not know it. 5 markers also, I've done it separately. HOTS questions are being covered over here. Go through them. So this is what I had to tell you guys, and make sure you do that. And yes. So Vinayak, so this is a thing that I have got previously. That is exactly why I'm stopping right over here. I understand your concern, but there might not be too many children who would want me. So please be there tomorrow. Sharp at 4:30 or something like that, I'll be coming live where I'll be solving complete biology. So be there for that particular live. I'll take it completely. I might even go at, long at night. So be ready for that. And thank you, my dear children. Wishing you all the best. Do not forget to hit the subscribe button. And I hope it's fine. Vinayak, I hope you'll forgive me for this. Yes, you have the resource. I'm giving you the resource. The same question is there in the 1 marker, in the MCQs. Okay, so please do that. Sanjeevani, thank you so much. You have done so well. I'm so proud of you, getting all the answers and all. And yes, take some rest and have some lunch. Similarly, same to you guys as well. Thank you. Lots of love. Take care. Miss you. And see you really soon for tomorrow's exam. So for tomorrow's live. Bye. Yes. Okay. Perfect. Bye.